Good morning, welcome. Thank you for joining us for day 2 of the Showcase Symposium. We are going to be getting started again at the top of the hour, which is 9 o'clock here on Pacific time. So while we wait for the top of the hour to arrive, please introduce yourself using the chat function here in Zoom, or if you're joining us on YouTube using the live chat. Please tell us where your name, your affiliation, and where you're calling in from, and tell us something that you're excited to be learning about today. Make sure you send those chats to all panelists and attendees so that everyone can see your responses. At any point during today's talks, you can ask our team talk and our next generation leaders speakers questions using the Q&A function here in Zoom. That's the one that looks like 2 speech bubbles. And make sure you include the speaker's name or the team talk title so that we know who that question is for. So you could say, hey, Caitlin, will these talks be recorded and posted? And the answer is that they are instantly being streamed to YouTube and they are available immediately. If you're having trouble here on Zoom getting connected, you have 2 options. You can watch on YouTube. You can click that link in the top left corner, view on custom live streaming surface. That's going to take you over to YouTube. Or you can try switching to your phone audio to minimize the strain on your internet. And here's an overview of today's agenda. We're going to start with a team talk by our MindScope mFISH team. We're going to have a couple of NGL speakers. We're going to have our final team talk from the Allen Institute on genomics, one more next generation leader, and then wrap up with some closing remarks from Alan Jones. So again, thank you everyone for joining us for day 2 out of 2 of our 2021 Showcase Symposium. My name is Caitlin Casimo. I lead training and education programming here at the Allen Institute, and I'm a member of the Showcase Committee. As we wait to get started at the top of the hour, which is 9 o'clock here on Pacific time, please use the Zoom chat function to introduce yourself. Let us know your name, your affiliation, where you're calling in from, and what you're excited to be learning about today. And make sure you're sending those messages to all panelists and attendees so that everyone can see your chat messages. But if you have any questions, please make sure to ask those using the Q&A function in Zoom. And make sure you include the speaker's name or talk title. If you're joining us on YouTube Live, there isn't a separate Q&A and chat function. There's all just the one chat, so you can just ask your questions there. We'll bring them over to Zoom so that those who are here in Zoom with our moderators will be able to see those questions. If you have questions after the fact also, you can visit our community forum at community.brain-map.org. We have a thread set up for yesterday's talks, and we'll have another thread set up for today's talks where you can ask follow-up questions, and you'll also be able to see the written responses provided by our speakers to questions that we didn't have time to answer out loud. So those responses are going to be available. If you are having trouble here in Zoom, you can try connecting on YouTube. Click that view on custom live streaming service button at the top left corner of Zoom. Or you can click the YouTube button on the login page that you used to get here to Zoom this morning. Or you can just go to our YouTube channel at youtube.com/Allen Institute, and you can find the live stream there. Or you can try switching to phone audio. Again, here is today's agenda, and this is going to bring me to our first item in the agenda. So I'm going to hand it over to our moderator for this morning, Jenny Close.
Thank you, Caitlin. Welcome everyone to day 2 of our annual, now virtual Showcase Symposium. My name is Jenny Close. I'm a scientist on the mFISH team in the Allen Institute for Brain Science, and I'll be your MC this morning. I am pleased to introduce our first next gen leader to present today, Cindy Poo. Cindy is actually one of our 2020 NGL class, but she wasn't able to join us for Showcase last year. So we're delighted to have her present today. She's a postdoctoral researcher at Champollion Research in Lisbon, Portugal, in the lab of Dr. Zachary Meenan. In her presentation, Cortical Circuits for Olfactory Behavior, she will discuss her work in the hierarchical circuits and interactions linking olfaction and spatial cognition, leading to the construction of an olfactory place map. Welcome, Cindy.
Okay, hi. All right, I hope you guys can hear me. Yeah, thank you very much for the Allen Institute for giving me this opportunity to share my work with you today, particularly since I missed the showcase last year. So I'm extra grateful to be here today. And I also have appreciated the land acknowledgements that have been part of the showcase this year. As it was mentioned, I live and work in Portugal, in Lisbon, Portugal. So while this is not indigenous lands, the Portuguese Empire has colonized and displaced indigenous populations across vast regions of South America. So I think that while the traditional land acknowledgement formula might not apply here, but I would just like to acknowledge the link between where I work now and recognize indigenous peoples of Brazil. Okay, so so for my talk today, I'm going to be telling you about cortical circuits for olfactory behavior. One of the most important ways in which animals understand and interact with the world is through chemosensation. So for example, by detecting volatile chemical molecules in the air through their olfactory system. So this is important for a wide range of ethologically important behaviors, ranging from locating valuable resources like food or shelter to avoiding predators, as well as mate selection. And my past and current work has focused on understanding the circuits and population dynamics that is the basis of olfactory behaviors. At the very end, I'll also like to tell you a little bit about my future plans moving forward, where I'm going to be looking at more naturalistic and flexible behaviors using odors and spatial and the spatial environment. Okay, so olfaction is not only important for rodents, which is my model organism, but the ability to detect chemicals in the environment is fundamentally guides the survival for a wide range of organisms. So something as simple as chemotaxis in bacteria to something that's more complex, such as foraging, memory-guided foraging in the honeybees. While this type of olfactory behavior or spatial behavior guided by odors is not thought of as being very prominent in humans, I think many of us can relate to the experience of a particular smell evoking a very powerful and vivid memory that transports us across space and time. So I think that this is revealing and intuitive for us to understand the fact that odors not only guide spatial behaviors in many organisms, but that in humans, there's also a privileged connection between olfaction and spatial cognition. Okay, so how do we perceive odors? How is the information about chemical volatiles in the environment? How does this information get into the brain? So, so in the rodent brain, the main cortical region devoted for odor information processing is the primary olfactory or piriform cortex, shown in purple here. Piriform is the first stage of cortical information processing, and neurons here receive direct afferent input from the olfactory bulb. And it is a spiking activity of ensembles of neurons here in piriform that is thought to be responsible for odor perception, discrimination, and recognition. And so in order to understand odor perception, discrimination, and ultimately behaviors that are associated with odor perception, we must first understand what leads to the spiking activity of neurons in piriform. And one of the things that, and this is one of the things that I looked at as part of my graduate work, that is, how can we understand the circuit components that these neurons are embedded in, because the circuit components that neurons are embedded in constrain the local computation and properties of representation in any particular brain region. So in piriform, the case of piriform, besides input coming in from the olfactory bulb, there are also a lot of cortical excitatory connections. So these are synapses that originate from within the cortex, and they can be recurrent between the piriform cortical neurons themselves, or long range coming from top-down top-down higher level cortical regions that is outside of the piriform cortex. So, and one of the really interesting things that we discovered during my graduate work is that even though piriform is a primary sensory region, these cortical synapses actually dominate the spike output of piriform neurons. So, and that these cortical synapses are also highly plastic. So this shows that, you know, information that's coming in from the bulb can be completely transformed in piriform cortex by these local recurrent and long-range top-down synapses. And whenever an odor molecule activates the olfactory pathway, these cortical synapses is very powerful in transforming the representation in piriform. So, you know, I mentioned that there's two different types of cortical synapses. There's the local recurrent and the long-range excitatory. There has been a large body of very interesting work showing how recurrent connections can support computations that are important for things like perceptual stability or associative learning. And this might be particularly relevant for piriform cortex, given the vast complexity and high dimensionality of the problem of odor recognition, because there's so many different types of molecules out there in the world. However, the other part of these cortical synapses, so the long-range excitatory connection, we know very little about. So this is kind of the starting point of my postdoc work, so my current work. This is what guided my current postdoc work, the question of what is the contribution of these long-range excitatory projections into piriform cortex, and how do these synapses actually contribute to the computation and the behavioral role for piriform cortex. Okay, so in looking at the top-down inputs to piriform, we noticed that, you know, it was very interesting that one of the major sources of these inputs is actually the hippocampal formation. So the hippocampal formation consists of the hippocampus proper, as well as the entorhinal cortices. And these regions, of course, not only famous for the discovery of place cells and grid cells and their role in spatial navigation, but also episodic memory. And very interestingly, they also share a developmental and evolutionary origin with piriform cortex, in that they're all allocortex. So this part of the brain that's very ancient in evolutionary origin and is largely conserved across a wide range of mammalian species. So all these things are really intriguing to us. And we consider the possibility that piriform cortex actually could be participating in spatial cognition. So to investigate this, we designed a new task where rats need to use both incoming odor information as well as their knowledge about the spatial environment to navigate on an elevated plus maze to obtain rewards. And then we recorded from interconnected olfactory and spatial memory centers in the brain. Okay, so this is kind of the setup of our task. Like I said, it's an elevated plus maze that is 1 meter by 1 meter in footprint. So at the end of each of these arms, there's a port that can deliver either a light cue, one of 4 possible odors, or a water reward. And the job of the rat is to initiate the trial following a light. And after initiation, they will get 1 of 4 odors. And then depending on the odor identity, the rat must choose one of the 4 ports to poke in again. And if they're correct, they'll receive a droplet of water. If they're incorrect, they'll go through a prolonged intertrial interval. So importantly, the correct choice, depends solely on the odor identity, but the odors can be presented at any one of the 4 odor ports. So the reason for this task design is to encourage animals to use the spatial environment, the allocentric, so world-centric reference frame to navigate instead of an egocentric or action sequence to solve the task, because this essentially breaks the contingency between action and goal. Okay, so what does the behavior look like? So here we see the plus maze is in a room with visible visual landmarks. This rat is here. He's going to poke in the north port, gets an odor, chooses the correct port, east in this case, because it was odor 3. And in the next trial, you see he gets the same odor now, but at a different sampling location. Oh, he almost got it wrong, but he got it. There's interesting stuff happening in the intersection, which I won't talk about today, but I'm very interested in exploring that later. And the next trial, he goes to the same sampling location for a different odor, and in this case, the correct choice was to go north. He got it correct. So after some training, the rats perform quite well in this task across different odor identities as well as sampling locations. So here, chance is 25% across all conditions because there's 4 of each possible choice. Okay, so like I was mentioning before, our intention was for the animals to use the world-centric reference frame to solve that task, meaning if I get odor 1, I go north, etc. However, as many of you who work with animals know that they're not always using the strategy that we intend them to use in solving the task. So how can we know what strategy rats are actually using, and if they're actually using both olfactory and spatial knowledge? So here, what we did is that we took advantage of the fact that we know animals largely have this win, stay, lose, shift strategy, meaning that given a reward on the previous trial, the current trial choice is biased in that, in other words, if animals were using an egocentric strategy in which they were keeping track of action sequences that led to reward, they'd be more likely to repeat the same action on the current trial given a previous reward. Similarly, if they're using allocentric strategy, where they're keeping track of reward locations, they'd be more likely to revisit rewarded locations. So when we did this, we found that animals were more likely to revisit previously rewarded locations, but not more likely to repeat the same action. So we think that in this task, right, and so this is just to summarize across all 4 different spatial port choices. So what this means is that indeed rats here are using odors and engaging in a spatial reference frame and using that information to navigate. Okay, so now we have our task. We targeted our chronically implanted tetra recordings to both the posterior region of piriform cortex as well as dorsal CA1. And we focused on the posterior part of piriform because this is a region where is a thought where the neurons, or this is a region where we were thought that projections from the hippocampal formation are enriched. Okay, so what do neurons in piriform look like when rats are performing this task? So I'm going to show you a couple of example cells first. So you can see here on the raster plot on the bottom and the PSTH on top here aligned to time of odor onset, that this is your standard odor selective neuron. It likes the yellow odor, has an increase in firing rate, but silent to the other 4 odors. However, now, if we look at the firing of the cell instead of across time, but aligned on XY position on the maze, so here, this is essentially a firing rate heat map, we see that we see 4 spots lighting up for the odor 4, meaning that the cell is selected for odor 4, but doesn't care about the sampling location. And here I'm showing you another example cell also recorded in piriform in the same session. You can see the cell similarly is recruited to fire in response to odor 4. However, if we look at the firing rate heat map across space, we see something very different. What we can clearly see is that activity to odor 4 is restricted only to the west port. And if we look across the other odors, we see that this cell actually doesn't care much about the identity of the odor, but for all odors is spatially selective to fire only at the west location. We also see cells that is a combination of the two. So for example, here, the cell is active to the blue odor only at the north port. We can summarize this by taking a decoding approach and ask how well a linear classifier, so here we're using SVM linear classifier, can read out odor identity or port location across the entire recorded population. And what we found was that both odor identity, which is in red here, and locations can be decoded from the population. So here we're randomly sampling increasing numbers of neurons for all the recorded neurons. And we can also take a sparse decoding approach where we considered the most informative neurons first. And you can see that much smaller population of neurons are required to reach high accuracy, but still, even in this case, locations are more prominently represented. So the decoding performance for locations is actually much better than odors here, which is quite surprising for us. And even more surprising was when we started to look at how the neural activity evolved across time. So here what we're doing is we're looking at simultaneously recorded populations only. And using the similar decoding approach, now we can look at the classification accuracy across time, where 0 again is odor onset. And of course, not surprising, odor identity only is available and therefore read out from the population above chance after time 0. But the location code is so that the decoder can actually successfully know where the animal is even before odors were available. Okay, so this means is that the place, the information about location where the animal is actually is independent of the olfactory drive, which was quite surprising to us. And it raised the question of whether this indeed is a spatial map or spatial representation of the environment, or perhaps given that it's locked, ramping up to time 0, maybe it's some kind of event-specific or predictive signal. So in order to kind of differentiate these possibilities, we looked at the neural activity across different task events in our trial. So we looked at the activity around odor sampling as the animal's approaching to goal or during the intertrial intervals. And in these 3 example cells, you can very clearly see that the most spatially informative neuron in cell 2 here, the spatial selectivity is actually consistent and persists across the different task epochs. And we can quantify this across the population by training a classifier on the activity during one epoch and testing it on the other epochs. Okay, so here on the x-axis, we have the cross-validated performance of the decoder trained on the initiation period. And on the y-axis is the performance of the same decoder on a different time window, so during the goal window. And you can see here each data point is a simultaneously recorded population. The size of the data points reflect the number of cells in each neural ensemble. The open ones are puriform, the gray ones are CA1 recordings. And we see that the spatial information is actually stable across these 2 different time epochs during the trial. And this is even true when we took the same approach and looked at the activity during the intertrial intervals. So that this means is that, so what this data is telling us is that indeed the spatial information we see could actually potentially be used as a way for animals to know where they are, because it's stable, independent of behavior states, and independent of the immediate sensory drive. Okay, so so far what I've shown you is like how similar the spatial information is in pure form to CA1. So we asked whether indeed we can find a functional relationship between CA1 and puriform. And so to do this, we looked at the coherence of spike timing of puriform cells to either the sniffing signal recorded with a thermal couple implanted in the nasal cavity, or to the local field potential recorded in CA1. So the coherence essentially is a way where we can measure ongoing functional connectivity between the puriform spikes, either to bottom-up sensory drive or potentially to the neural activity in the hippocampal network. So what we did is we looked at this coherence dependent on the single cell decoding accuracy for either odor or spatial location. And what we found was that if we look at these coherence plots of time here, again, 0 is time of odor onset, and the y-axis is frequency, that we see for the odor informative cells on the left column here, these guys are strongly coupled to the respiratory signal, whereas the location informative cells are not very much. And right, so the colored pixels here are frequency time bands in which the coherence is statistically significant. Okay, so and when we look at the CA1 LFP, we see something very different. And we see what we see for the location cells on the right here is that these guys actually are quite strongly coupled to the CA1 LFP at theta band frequency, and only coupled, transiently coupled to the respiration after odor onset. So this actually was, and we don't go into it very much here, but I think this is really fascinating because this potentially reflects a transient coupling between the sniffing and the theta frequency in hippocampus, which is something that is known about sniffing and theta. Okay, so what this implies is that in puriform, there are potentially different cell types or different subnetworks of neurons, where some neurons are more coupled to the sensory drive and some neurons are more coupled to the hippocampal network. And all these neurons are intermingled, okay, intermingled within puriform. So, and you know, the thing that we were curious about is, what does this mean about puriform in terms of its relevance? Are these cells just passively reflecting a copy of what's going on in the hippocampus and they're just inheriting it? Or is there something different that's happening, right? So, and we looked at this, and we looked, something interesting happened when we started looking at the spatial representation of puriform neurons away from the odor port location. So as I was mentioning to you before, we have this elevated plasma, so we can look at essentially the firing of CA1 and puriform cells, not just at the port locations, but also all along the corridor. And when we do this, what we see is that, so for each row now here is a single cell that's recorded in CA1 or puriform, and the increase in brightness reflects increase in firing rate for the cell. And on the x-axis is distance to port for all 4 of the arms, going from the best arm, so the arm that has the highest firing rate, to the arm with the lowest firing rate. And you can see very clearly is that CA1 cells kind of tile the entire length of the arm, whereas the puriform and the spatially informative puriform neurons cluster around the ports, okay? So, so we can quantify this, of course, using a similar decoding approach. And what we see is that our ability to decode location from the puriform population drops off significantly as animals move away from the odor ports. Okay, so what this means is that, you know, this uneven representation of the maze by puriform suggests that these spatial maps is actually, you know, these spatial maps are potentially learned and reflect locations that are behaviorally relevant for the animal. What it also suggests is that there is actually more than one map for space in the brain, and that, you know, brings us to the part of my work which is more ongoing, and it's a collaborative collaborative effort where we asked the question, how can we start to compare or start thinking about multiple different cognitive maps in the brain, and how can we compare and measure in a principled way what these different neural populations are representing. Okay, so right, so another way of saying this is, if we have a neural population 1 that cares about these locations and 2 that cares about these locations, how can we compare them and understand how relevant they are for behavior, and which one of these maps is potentially more optimal representation with respect to a particular task. So my collaborators, Mattia Bergomi and Pietro Vertecci, have been developing a formal method for this goal. What they do is that they can recreate our experimental task as a reinforcement learning environment and design an artificial agent that can be trained to solve this task optimally. And then they use a method which they've developed based on topology called generalized persistence analysis. And using this method, they can compute and rank relevance of different neural states or clusters of neural states of the optimal agent according to how relevant these neural states or clusters of states are for the task. So the key here is that this method allows us to formally quantify the distance between neural representations from each other, as well as from the one that's used by the optimal agent. And the goal is to work towards understanding computations in both biological and artificial agents. That's important for cognition and flexible behavior. Okay, so and one interesting thing about this method, of course, is that it can be used for tasks that are both spatial and abstract. And this is important because, even though I've only been discussing what hippocampus is good for in the context of spatial navigation, we know that the hippocampus cares about many more things besides just where the animal is at a given instant. Okay, so and one of these things is that it cares also about the relational and structural knowledge of the environment. So for example, the relationship between these two vehicles, these three different types of modes of transportation. So recent interesting work in showing suggests that the types of representations that's useful for in a navigation context may actually be an instance of a general coding mechanism of the brain to organize abstract and relational knowledge of all kinds. Okay, and what this means for puriform is that what we find could be relevant not just in the context of spatial navigation, but maybe it reflects the involvement or the role for puriform in other higher level functions associated with odors. So going forward, I think I'll skip the slide because I'm a little bit short on time. So going forward, I'm really excited to use the olfactory and spatial memory centers in rodents as a model system towards understanding the dynamic relationship between sensory and memory centers. And particularly, I'm interested in naturalistic behaviors such as foraging or navigation, where there is a dynamic updating of different goals or needs of the animal. And I'm interested in this at generally in these three different levels. So what is the computation, the inter-areal dynamics and behavioral function, as well as the circuit mechanisms and questions. And I'm interested in questions such as, what are the computations and transformations underlying flexible behavior? How do these interconnected regions contribute in different ways? And what are the cell type specific diagrams for, these olfactory and spatial regions? So experimentally, I'm interested in approaching these questions using both freely moving animals because it has an advantage of really rapid task acquisition, and where we can look at sequences of natural behavior and record chronically during learning. And also I've been developing a head-fixed olfactory virtual reality where we have, while we don't have as dynamic of a naturalistic behavior, we have very good control over the precise spatial and olfactory environment of the animal. And we can, and it's amenable to recordings across different interconnected brain regions. And lastly, even though I only discussed piriform cortex and the olfactory bulb today, as part of the olfactory system, we know that the olfactory bulb actually targets many more brain regions besides the piriform. So, the anterior olfactory nucleus and many more. How these different brain regions are connected, what cell types are there is actually very poorly understood. And I think, olfaction is relatively underexplored as compared to other sensory modalities. So I'm really interested in building a cell type specific circuit diagram across these olfactory regions with the goal of understanding how they work together to guide behavior. Okay, so I would like to thank everybody that has been involved in this work. I did the first part, the circuit part that I mentioned briefly at the beginning in the lab of Jeff Isaacson at UCSD. I've been doing my postdoc in Zach Maynon's lab at the Champagne-Limau. The main project was a collaboration with Gautam and Nico, who were instrumental in helping me think and develop the task. And as well, I would like to thank current collaborators, Mattia and Pietro. The hardware and animal care facility platform also has been huge in supporting me throughout the experimental phases. And yeah, I would like to thank also our funding sources and your attention. So yeah, I'll take questions.
A great talk, Cindy. Thanks a lot. As a reminder, if you have questions for our speakers today, our next gen leaders throughout the day, please submit them via the Q&A panel and include the name of the presenter that your question is for. We do have time for a few of those, and a few questions have come in. So the first one is, would you expect if you did another odor behavior test without spatial element, for example, just an odor tone test, would you still see the same level of activity in the piriform?
Yeah, I think that's a great question. I'm also very curious. I think that, I'm going to kind of, have my cake and eat it too. So I think that, yes, we're going to see also some, interesting associations that are learned signals that reflect learning in piriform in non-spatial tasks. But I do think that there's something that is privileged between piriform and spatial memory centers. And this is not just, and this is from the anatomy, also from, developmental origins and, shared cell types and connectivity. So I think while, we can think of piriform as perhaps generally dedicated to learning things that could be associated with odors, but perhaps that spatial information could be privileged in some way. So this is my guess.
Cool. So there are a few more questions in the Q&A for Cindy to get to. They look like really good questions. So I'll just thank you again for a great talk. And it's great to hear you speak this year. Thanks.
Thank you.
So now we're going to move on to our first project talk of the day from the MindScope program, the mFISH project, the MindScope program's mFISH project team. Titled Connecting Functional and Transcriptomic Neuronal Types. So the MindScope program has long been investigating neuronal functional physiology, particularly in visual processing. And these two new projects, they are now linking transcriptomic cell types and those identified in the Allen Institute for Brain Science cell types work to their functional types. So take it away.
Good morning. I'm Saskia de Vries. I will be launching our talk on connecting functional and transcriptomic neuronal types. So we have two projects in the phase 4 of the MindScope program that are looking to connect functional physiology with transcriptomic cell identities. And Pete and I are going to start by giving you kind of the context of those different projects before the rest of the team will launch in and tell you more about the details of how we're integrating these two modalities. So this work really is rooted in work that's been going on at the Institute for the last 10 years. So first in the MindScope program, we've been building these in vivo physiology pipelines that allow us to record the activity from large populations of neurons. In particular, we have a 2-photon calcium imaging pipeline where you can see here the activity of hundreds of neurons that we image at the same time. And different cells you see light up at different times, responding to different features of the stimulus or different behavioral phenomenon going on during these sessions. So on the right, you see just the activity of these different neurons, and again, just highlighting the diverse and heterogeneous responses that we're seeing. Now, at the same time, in the cell types program in brain science, there's been a lot of work establishing transcriptomic identities of different neuronal cell types, and then building in the imaging team a platform that enables spatial transcriptomics, leveraging a light sheet imaging to identify the genes that are being expressed in different cells in a piece of tissue. And what we're looking to do in a couple of the projects that we have in MindScope is to link the transcriptomic identity of cells that we can determine based off of these, these gene profiles to their diverse physiological functions. So there's two projects. One is the V1 mFISH project. I'm going to give you a little bit of the context of the questions that we're looking at there, and then I'll hand it over to Pete, who can tell you about some of the goals for the Learning in mFISH project. So our V1 mFISH project really is rooted in a lot of the questions that came out of our visual coding data set regarding the heterogeneity of the different types of responses that we saw. And we found different types of responses and that didn't map very cleanly onto, say, the Cree lines that we were using, but we know that these Cree lines aren't very specific to transcriptomic cell types. And so particularly for the excitatory cells that we're looking at, we really want to dig deeper into this question of whether these heterogeneous visual responses map more cleanly onto transcriptomically distinct cell types. At the same time, we also are looking at questions about the inhibitory interneurons. And so in our previous work, we found that the balance of inhibitory interneurons, particularly the VIP and somatostatin neurons, regulate the cortical dynamics, so that when VIP cells are highly active, there's a high gain regime in the cortex, whereas the somatostatin cells, we see there's a low gain regime that's enhancing the stability, maintaining the stability of the recurrent excitation in that cortical circuit. And we found evidence that this balance and the interaction between the VIP and somatostatin cells seems to really depend on the stimulus and behavioral context of during the experiments. And so what we want to be able to do here is by imaging these cell types simultaneously, but then identifying them after the fact, we can then look more closely at the interactions between these different populations and how they depend on the stimulus context. So now I'll turn it over to Pete, and he will tell you more about the goals of the learning project.
Thanks, Saskia. In our project, we're interested in understanding how distinct learning processes restructure the activity of excitatory and inhibitory cell types in both the visual and frontal cortex. So to do this, we are developing a staged learning task that progresses mice through a series of visually guided behaviors, from basic associative learning to more complex operant learning, and finally, extinction learning. We'll add strategically timed longitudinal 2-photon calcium imaging of a set of cells in order to assess the functional changes that occur in cortical neurons across these different types of learning. Once the in vivo portion of the experiment is complete, we'll use spatial transcriptomics to map the cell type identity of the cells we imaged during this task. So both of these projects require linking transcriptomic identity to physiological function. And to do this, we've been working on integrating these two separate experimental workflows, the Allen Brain Observatory and the Lightsheet mFISH pipeline. And specifically, we focused our recent efforts on 3 key integration points. The first step in linking these pipelines is to accurately isolate the image tissue and prepare it for mFISH workflow. The second key integration point involves co-registering the cells of the mFISH volume back onto the functional 2-photon imaging volume. And lastly, the final integration step is to combine all the data streams collected in these two pipelines with the goal of assigning cell type identifications to the recorded neurons, thereby allowing for functional characterization of transcriptomically identified cell types. And it's important to note that developing all of these integration steps has been done in parallel, with the final goal of pulling them all together in the coming months. So for today's talk, we're going to highlight some of the key accomplishments that we've made with these pipelines and their integration steps. First, Shauna will describe the details of our in vivo imaging experiments and some of the modifications that we've made to facilitate this new workflow. Next, June and Erica will share how they are isolating and preparing the functionally imaged tissue for mFISH processing. Mike will then tell you about some of the progress that the imaging team has made to most efficiently identify cell types in our tissue samples. And lastly, Alan will show you how we plan to co-register the mFISH volumes with the previously collected imaging volumes. So with that, I will pass it off to Shauna.
All right, thank you, Pete. So as was mentioned, we're using the existing 2-photon pipeline from the Allen Brain Observatory for these projects. We have a suite of transgenic mice that allow us to image inhibitory and excitatory neural populations. After these mice have reached their designated age and weight, we perform surgery to place a cranial window and titanium head frame over their visual cortex. We then perform intrinsic signal imaging, which generates a retinotopic map, which we use for targeting in 2-photon. For this project, we're using a multiplane scope called DeepScope, which uses a diffractive spatial light modulator, which shapes the incoming 2-photon beam and allows us to image 6 separate planes in the same cortical column in V1, layer 2-3. The data outputs for this project look similar to past Allen Brain Observatory data sets, including targeting in V1, stimuli such as natural movies and drifting gratings, behavior, body, eye, face recordings, as well as the 2P recordings from the 6 imaging planes. But we've also made modifications to our data set to help facilitate downstream steps. For example, we have an 100 micrometer epi surface stack and an 100 micrometer 2P surface stack, which help locate the field of view in the light sheet, which June and Erica will speak more to. We also have a detailed cortical stack of the entire column, which helps facilitate co-registration. After we're done with the tissue, it moves on to sectioning and embedding, which June and Erica will tell you more about.
Thank you, Shauna. The next step after in vivo imaging is to collect the tissue and prepare it for the following mFISH experiment. Because this step links the in vivo imaging and the ex vivo imaging modalities, we want to optimize these steps to make the downstream alignment and co-registration as simple as possible. For that reason, we found the chronosection method was not ideal because our 2-photon imaging had high resolution in tangential dimensions, but not in the chrono dimensions. So we adapted a tangential section method developed in one of our other projects, which had just been published. With this method, we can align the tangential surface with tangential surface. As you can see here, this method showed promising results for the alignment between the in vivo and in vitro imaging modalities. How did we do that? We took advantage of the fact that the glass cranial window already flattened the cortical surface of interest so that we can mount our fixed brain with this flattened surface against a glass slide. Then we use a blade parallel to this surface to cut one thick section with a thickness of about 400 microns. Because this piece of cortex is slightly compressed by the glass window, and we found this thickness can cover almost entire cortical tabs, which is good because our mFISH team has only to deal with one section per mouse. However, there's another problem, which is this roundish symmetrical section shape is not good for us to keep track of the tissue orientation, which is important for the initial coarse alignment, which will help the alignment and the co-registration at a single cell resolution. To overcome this challenge, we worked with our manufacturing and processing engineering department to develop a biopsy punch that can cut out a asymmetrical trapezoid shape out of this 5mm window. We make this trapezoid shape much larger than our 2-photon field of view, so we can make sure that the 2-photon view is completely within this shape. To make this method even more robust, we use a black ink to dye the tip of this trapezoid so that the tissue orientation can be tracked easily in the following steps, as Erica will show you next.
Thanks, June. The next step in this process is to embed the tissue in a hydrogel. Hydrogels are swellable and expandable, leading to optically transparent samples, which increases the imaging resolution and the signal-to-noise ratio. We also use a chemical called mFISH, which preserves RNA in the tissue. Here's an example of a piece of mouse tissue going through gelation. First, the sample is placed in a gelation chamber, which keeps the tissue flat, and then it's baked at 37 degrees Celsius for 2 hours. After gelling, the gel is trimmed into an asymmetrical shape, which aids in orientation. Then the tissue gel is digested with proteinase K. Finally, the gel is cleared and expanded. You can see the fiducial mark that was placed during sectioning by June. Next, Mike will talk about mFISH and imaging.
Thanks, Erica. Our overall goal at this point in the pipeline is to supplement the live cell imaging data with a data set that can map each cell to a transcriptomically defined type. An important thing to recall at this time as well is that this population of cells resides within a 3-dimensional volume that may span many cortical layers of depth within the tissue. Now, this would be difficult to capture via thinly sectioned tissue mounted onto slides. We accomplish this goal, however, by fluorescence in situ hybridization of probes within these thick but clear sections that June and Erica have described. Here I'm showing an example where Cree-driven expression of fluorescent protein in green is supplemented with labeling of 3 specific RNA species utilizing different excitation and emission channels, here in yellow, in blue, and in magenta. Lastly, we use a 5th channel to include a more generic label of all cell bodies, which can be used for image segmentation and registration. This last step is important for extending the number of genes probed beyond the available fluorescent channels by repeatedly staining and imaging the tissue. So how many genes are necessary for mapping a transcriptomically defined cell type? Looking at all cell types, a large number of genes would seem to be necessary. Fortunately, concentrating on specific classes or subclasses can greatly reduce this requirement to a dozen or so genes. Here I'm showing how the confidence of mapping single-cell RNA-seq data back onto itself increases with the number of genes probed. For inhibitory cells containing GAD2, 15 genes gets us to a level of 67%. While looking more specifically at SST subclasses, 15 genes reaches an 80% confidence level, which is near the best case. In this way, we can maximize the information gathered by judicious choice of genes within the class of cells targeted during live 2-photon imaging. In order to gather this data, cleared tissue is imaged on a light sheet microscope, which decouples the excitation and emission pathways. By optically sectioning the tissue with excitation light, large volumes can be acquired by scanning across areas of the sample. Here you see a top-down view of the tissue where these horizontal bands are individual stage positions that have been scanned and tiled. Below is a view through the depth, showing an additional layer of data collected at a deeper level. And with some computational efforts, a Z-stack through the entire thickness is obtained, here shown in the inset. Finally, the result is an image volume where we can clearly see cells belonging to some Cree-driven marker line, as well as cells additionally labeled by their expression of a few chosen genes, here SST in yellow and parvalumin in magenta. So now that we have image volumes of both transcriptomic expression and electrical activity that contain the same population of cells, these two data sets need to be placed into a common coordinate system to form a complete profile from both modalities. And now Alan will describe in detail how this can be achieved.
Thanks, Mike. As Pete mentioned earlier in the talk, one of our goals is to link data collected as part of our Brain Observatory 2-photon pipeline, which Shauna described to you earlier, to transcriptomic information obtained through the Lightsheet mFISH pipeline, which Mike just outlined. This is where co-registration comes in. Here, we seek to put data from these two imaging modalities into a common coordinate system, thereby allowing us to perform integrated analyses which leverage both functional and transcriptomic information. To accomplish this, we utilize two imaging volumes. The first of these is a detailed cortical stack collected during 2-photon imaging. As Shauna mentioned earlier, this is one of the modifications we made to the Brain Observatory pipeline specifically for this project. The second imaging volume is a similar set of images collected during the Lightsheet mFISH pipeline. Our goal then is to take cells in the 2-photon volume and find the corresponding cells in the Lightsheet volume. To do this, we use a two-stage approach. In the first stage, imaging volumes are converted into a set of three-dimensional points using segmentation. In the second stage, 2-photon and Lightsheet point clouds are aligned such that they are in a common coordinate system. This allows us to match points between the two volumes. In the segmentation step, we use a convolutional neural network to transform our image volumes, shown on the left, into a set of three-dimensional segmentation masks, shown on the right, where each mask labels a single cell. We can then compute the center of mass of each of these masks to obtain a three-dimensional point cloud. Here, each point represents the location of a single cell in the imaged volume. Once we are finished with segmentation, we then have two sets of point clouds, one for each of our imaged volumes. We now want to align these point clouds so that they are in a common coordinate system. To do this, we use a technique called iterative closest point, which seeks to identify a rigid transformation from one point cloud to another that minimizes the distance between points in the two clouds. To give you a little intuition for how this works, considering the following example where we have two sets of points, one representing the 2-photon volume in green, and the other representing the Lightsheet volume in purple. If we were to plot these two sets of points in the same coordinate system, we can see that they are misaligned. To fix this, we first match points between the two sets such that each point in one volume is matched with the closest point in the other volume. Once we have done this, we then compute a rigid transformation that minimizes the distance between sets of matched points. This process is repeated until convergence. Here is an example of what this looks like for one of our pilot datasets. Each panel here shows a two-dimensional view of the same three-dimensional volume. Green points represent the location of cells in the 2-photon volume, and purple points represent the location of cells in the Lightsheet volume. Matched points are connected with black lines. As you can see, in this example, the iterative closest point technique does a good job of aligning these two sets of points, such that the distance between matched pairs of points is small. To get another sense for how well our co-registration process is working, we can look at the Lightsheet image volume, resliced such that it is in the same coordinate system as the 2-photon volume. For this example, you can see that the location of cells in the 2-photon volume on the left is qualitatively similar to the location of the cells in the aligned Lightsheet volume on the right. For example, this particular constellation of 6 cells in the 2-photon volume is in approximately the same location in the Lightsheet volume, thus confirming that these two sets of volumes are aligned. So, bringing everything together, now that we have put these two image volumes into the same coordinate system, we can enable integrated analyses which links functional properties from the 2-photon pipeline to transcriptomic information from the mFISH pipeline. And with that, I'd like to turn things over to Anton, who will provide some concluding remarks.
Thank you, Alan. Now to the conclusions. So in this project, we are attempting to connect transcriptomic cell types in the mouse cortex with their in vivo function. When the different types of work that you just heard about come together, we'll be in a great position to gain deep insights into the roles of these transcriptomic cell types in visual processing, regulation of cortical dynamics, and learning. Not only that, but we will also make all of this data publicly available, and that will constitute an atlas of cell types reference to their function. Finally, we will be able to connect this data to the other great data sets at the Allen Institute, such as the PatchSeq dataset, where individual neurons are characterized transcriptomically, morphologically, and electrophysiologically, or the electron microscopy dataset, where a very similar visual activity is collected in vivo, and then electron microscopy reconstructions allow to characterize morphology and connectivity of individual cells. So together, this data will open a new window into the roles of cell types in brain activity and function, and will provide an excellent resource for the community for future experimental and theoretical work. Over to you, Marina.
Thanks, Anton. So as you've seen today, connecting the 2-photon brain observatory for in vivo functional measurement with the Lightsheet mFISH pipeline for spatial transcriptomics involves extensive coordination across many teams and individuals. And we would like to thank everyone who's been working together to make this science possible. From our extremely talented research associates who care for the mice and collect lots of high quality data, to the engineers who build and maintain our experimental and data collection pipelines, the scientists designing experiments and analyzing data, and the project managers who keep us all coordinated and moving in the same direction. We're grateful to everyone involved, and thank you for your passion and dedication to big open team science. Thank you all for your attention.
Very interesting talk. Thank you for that MindScope mFISH team. So we do have a question or two to answer. One from Christina Kim, Alan and the rest of the team. So awesome to see the advancements in the co-registration. I was wondering what the rate of dropout is when you identify cell points across the two data sets. Within the same approximate volume, if you segment 100 cells in the 2P stack, do you also find 100 cells in that same area in the post-clearing stack? I also had the same question, so I'm looking forward to hearing what happens there.
Sure, I probably can give a non-satisfying answer. We're still in the early stages of kind of linking these pipelines together, and so really that requires having a lot of data sets that we have both the mFISH and the 2B imaging, and right now we only have maybe one very early preliminary data set where we have both of those sources. So I guess the non-satisfying answer is to be determined. So it's something that's on my mind at least, and I hope to have a better sense of that moving forward.
Okay. I've got another one, Saskia and colleagues, who's from Eric Shea Brown. Awesome talk. Do you think new insights into, say, the VIP, SST, or other cell type interactions will arise from the fact that these will be simultaneously imaged and separated in single recordings versus serially, if I'm understanding that right?
Yeah, absolutely. This is, I think, one of the big motivations for doing this. There's kind of two components. There's simultaneously imaging different populations and then being able to identify different transcriptomically defined subtypes and see differences within, say, VIP cells of the different subtypes of VIP cells. But we absolutely expect that we're going to learn more about the interactions of these different classes of neurons through this simultaneous imaging and then being able to identify them after the fact.
Okay. We have another question from Stephanie Seaman. Do you have any predictions about what cell subclasses might show different functional responses when you split down to the T-type level? No predictions.
Well, if I may take it, I think it's too early to tell. There are some indications in data from Saskia and others that there is heterogeneity among VIP and SST populations. So it's possible that we will see that mapping onto the T types, but it's not entirely clear yet. So I think we'll see what happens.
Cool. I had one real quick technical question. What's the factor of expansion that you get when you expand the kind of interested in chemistry related stuff with the making the brains into other things? Do you guys know how much bigger the plug gets?
Are we talking about expanding the hydrogel, Jenny?
Yeah. Like how much bigger does the brain get when you expand it?
It expands by a factor of 2.
Okay, cool. That's pretty big. Okay, so Nick Jorstad, okay, Mike's I guess typing in the answer, but I thought I'd read it anyway. Great talk. Are you able to extract morphological neuronal features from imaging in this pipeline? Sorry, Mike, I didn't mean to.
No, that's okay. So I think the answer is probably not. It's probably possible to get some fairly rough morphological features, but this image data doesn't really have the resolution to trace projections in the long range.
Okay. Well, we're actually, I think, a little bit ahead of schedule, so maybe we can take like a 2-minute break and be back at 10:05 for our next talk by Tsai.
Thank you.
Thanks, mFISH team. Hi, everyone. Welcome back. Hopefully you had a chance to have a little break. Our next speaker is 2021 Next Gen leader Tsai. Jess is a postdoc in the Department of Pediatric Oncology at the Dana-Farber Cancer Institute in the Laboratorio Petiti Bandopayai. She is also an attending physician in pediatric hematology, oncology, and stem cell transplant at the Dana-Farber Boston Children's Hospital Center and Blood Disorder Center. The presentation today is titled Transcription Factor Hijacking in Pediatric Gliomas. She'll present her research on the role of the transcription factor FOXR2 in diffuse intrinsic pontine gliomas and other cancers and the genetic and epigenetic mechanisms underlying its pathological expression. Welcome, Jess.
Thank you, Jenny. Thank you so much for the introduction. I'm really excited to be part of this year's NGL cohort. I just wanted to start and acknowledge that I live and work on the ancestral land of the Pawtucket, Massachusetts, Nipmuc, and Wampanoag tribal nations in Boston. I'm a neuroscientist by training and sort of accidentally became a cancer biologist. And I'm also a pediatric oncologist. And very broadly, I'm interested in how developmental pathways are hijacked in pediatric cancers to initiate oncogenesis. And specifically, I'm really interested in delineating how oncogenes are activated in pediatric brain tumors through both genetic and epigenetic mechanisms by using functional, genomic, transcriptomic, and epigenomic approaches. And I really want to emphasize that the project I'm going to tell you about today is really the product of collaboration across multiple research groups, across multiple countries, and simply would not have been possible without such a team effort, especially with getting really precious pediatric cancer samples, which are often hard to come by. So a quick roadmap of the talk today. I'm going to take you on what I think is a fun journey through one of my projects that's examining this transcription factor called FOXR2 and how it behaves as an oncogene to mediate oncogenesis, not only actually in pediatric brain tumors, but across a number of adult and pediatric human cancers. So I'll give you a brief introduction to how this project started and try to lend you a little bit of the clinical context that I think about when I see my patients, and also really the context that drives a lot of my research. And then I'll walk you through these questions, which are really focused on kind of dissecting apart how FOXR2 acts as an oncogene, including the extent to which it is expressed across human cancers, how its expression is actually activated. What I'll show you is actually normally in most postnatal tissues, FOXR2 expression is absent. And then I'll show you some functional data looking at necessity and sufficiency for oncogenesis, both in vitro and in vivo in some of our tumor models. And I think the big question, which we're still working on, is really how does FOXR2 exert its oncogenic effects mechanistically? So amongst pediatric cancers, brain and central nervous system tumors are actually the most common pediatric solid tumor. And then the second most common cancer, really second only to acute lymphoblastic leukemia. So you can see brain and CNS tumors are this red line, and this is over time from 1975 to 2010. And I always think this is really interesting. This inflection point in the 1980s actually reflects the commercial availability of magnetic resonance imaging for diagnosis for brain tumors. Unfortunately, CNS tumors are also the leading cause of pediatric cancer death. So over time, we've gotten a lot better at treating leukemias, especially in children. And while we've gotten a little bit better at treating brain and CNS tumors, we really need to do better. And the treatments for these patients are often really just limping them along without much curative intent. So I think in my mind, this is like a prime opportunity for all of the genomic data and transcriptomic data that we've been accumulating from tumors to really look at how can we target these tumors in better ways. Amongst pediatric brain tumors, I'm very interested in a type of pediatric high-grade glioma called diffuse intrinsic pontine glioma, or DIPG. These are terrible tumors. They're fatal pediatric brainstem tumors. You can see this tumor infiltrating the pons. And the median survival from diagnosis for this tumor is 12 months. While patients may be treated with radiation or chemotherapy, inevitably these cancers are fatal. And actually, we know a lot about the mutational landscape of DIPGs, and they're actually very well characterized. And DIPGs specifically are characterized by point mutations in histone 3, the K27M mutation. And while these mutations are thought to be critical initiating events in the tumors, the mutations alone are actually insufficient for tumor formation and require at least one additional genetic alteration for tumor formation. So amongst pediatric high-grade gliomas, the somatic mutational landscape is actually extremely well understood. That was characterized in this paper in 2017 that looked at the mutational landscape of 1,000 pediatric high-grade gliomas, including DIPG. However, the point of this is not to show you all the different alterations. The point of it is that the majority of these tumors actually underwent whole exome sequencing, and therefore the structural variant rearrangement landscape of these tumors was very poorly characterized due to lack of whole genome sequencing. Our groups at Dana-Farber are very fortunate to have access to nearly 200 pediatric high-grade glioma samples from patients prior to initiation of any treatment, including radiation. And all of these patients, their samples have undergone whole genome sequencing as well as RNA sequencing. So this has been sort of a way for us to get at this question of whether there's recurrent structural variants in these tumors. This is work that's been done with our group. This is a paper that was recently submitted and is actually available as a preprint online if you want to check it out. But what we found is that actually 10% of DIPGs have this recurrent structural variant that actually amplifies a MYC enhancer. So this is chromosome 8 here. MYC is highlighted in yellow. And this gene right here is actually a long non-coding RNA called CCDC26. And each of these blue tracks is actually a different tumor sample. And what you see is that they all have this recurrent amplification. Some of them are simple, some of them are more complex, which we won't get into today. But all of them have this structural rearrangement in this MYC enhancer. And this was pretty surprising and really led me to ask, what are other mechanisms of activating MYC? We actually know that MYC is the most commonly amplified oncogene in the cancer genome. There are many, many large-scale whole genome sequencing papers from TCGA showing this. But while this is the most commonly amplified oncogene, focal MYC amplifications account for a very, very small percentage of these brain tumors in children. So there must be other mechanisms of activating this oncogene besides simple genomic amplifications. And when we actually look at our cohort, it turns out that alternative mechanisms of activating MYC in these brain tumors actually account for nearly a quarter of DIPGs. And this sort of commute plot shows essentially every gray bar is a different tumor and the alterations are on the left. And this is how I stumbled upon this transcription factor FOXR2, which I'll tell you more about. It turns out that what's known about FOXR2 is that it's a fork-head transcription factor. So it's part of this very large FOX family of transcription factors that has a fork-head DNA binding domain. And what's interesting about FOXR2 is that it's known to stabilize MYC. What's also interesting is that all the tumors that have expression of FOXR2 actually do not have focal amplifications in MYC or MYC-N, suggesting that there's some functional redundancy. And so what this work really began as really just trying to understand DIPGs, this fatal pediatric cancer, and identify novel drivers. But I will show you that it has since expanded to really understanding FOXR2 as a pan-cancer oncogene. So this is some data from TCGA. But we've actually analyzed over 10,000 cancers, both adult and pediatric, using a number of different data sets. So TCGA, we've used the Cancer Cell Line Encyclopedia. We've had access to the INFORM consortium through our German collaborators, which is a very, very large pediatric cancer cohort of tumors that also undergo whole genome and RNA sequencing. And then we've also have access to some Dana-Farber samples in addition to samples from St. Jude. And what I'm showing you is just a subtype, some cancer subtypes that show FOXR2 expression. And what we find in that over 10,000 cancers that 70% of cancer subtypes have at least a tumor that shows aberrant FOXR2 expression. And actually almost 10% of individual cancers in this large cohort have aberrant expression of FOXR2. And this includes actually some fairly common adult cancers like melanoma, non-small cell lung cancers, and endometrial cancers. You see something very, very similar in the pediatric cancers. This is data from the INFORM consortium that FOXR2 is aberrantly expressed in neuroblastomas and high-grade gliomas, and actually some of the bone and soft tissue sarcomas that we see in kids as well. So that's all well and good, but why is that interesting? It's interesting because FOXR2 is actually normally not expressed. This is data from GTEx, from postnatal adult tissue, just showing you some lineages, including brain, breast, colon, lung, and skin, where there's really no expression of FOXR2. The only tissue that expresses FOXR2 postnatally is testis. And I'm not showing you this here, but we've also interrogated a number of mouse and human single-cell data sets to try to find additional cell types that may express FOXR2 and really haven't found anything besides testis. We wanted to look in further analysis at actually a subset of these FOXR2 expressing tumors for which we had access to both whole genome and RNA-seq data. And what's interesting is that we found that a subset of these tumors actually activate FOXR2 through a structural variant mechanism, whereby an enhancer is actually fused to FOXR2 to activate its expression. But as you can see here, the structural variants are in brown. This is really only a subset of FOXR2 expressing tumors. And for the remaining tumors, I actually was unable to identify a structural variant, a rearrangement, or a somatic SNB. And so we sort of went back to the drawing board. So the most simple thing is just to look at FOXR2 in reference sequences, in reference genomes. So if you look at any reference sequence, FOXR2 is 1 exon. I'm just showing you its upstream gene USP51 for reference. So we decided to look in RNA-seq from testis, thinking that that's the only tissue that actually we find that expresses FOXR2 normally. And what you see in testis is that there's a number of varying mRNA structures that have many, many different transcription start sites that are very different from the annotated FOXR2 exon that's in reference sequences. So these are all numbered backwards from 1, exon 1 is the reference sequence, and numbered -1 to -6 backwards. So you can see there's actually a transcription start site here, exon -3, there's one at exon -6, there's one at exon -5. And this is just a non-FOXR2 expressing cancer to show you as a control. So we sort of reason that perhaps these exon structures, these alternate transcription start sites, are actually the novel promoter and the real promoter for FOXR2. And indeed, that actually seems to be the case. We also looked in our different types of cancers, and we find that these similar exon structures also exist across a number of different lineages in cancers. Even amongst the structural variants, the breakpoints actually occur at these different exons. So we reasoned that exon -3 could be a FOXR2 promoter, and to do this, we did some chip sequencing. So on the top in black is just RNA-seq, which you've already seen, but this is a FOXR2 expressing cancer. A non-FOXR2 expressing cancer you see clearly has no expression. And then testis, which you see this exon -3 really nicely matches up with our cancers. And then we did H3K4 trimethyl ChIP-seq as well as H3K27 acetyl, which are promoter and enhancer marks respectively. And you can see that when we call peaks, there's a very, very nice peak that localizes on exon -3, suggesting that this is actually a FOXR2 promoter. I've done a little bit of additional motif analysis on this specific sequence. So if you take the exon -3 sequence and actually probe for factors that are predicted to bind this interval using a database called the Systrome database, this has almost 13,000 previously published ChIP-seq data sets. What's interesting is that this identified a number of different transcriptional complex members, again, suggesting that this is a promoter site. And what's also very interesting is that all of this ChIP-seq cell lines that were pulled out from this analysis are actually all cell lines that are known to express FOXR2. To kind of get at this question more specifically, is the promoter really required for FOXR2 expression? We ended up using CRISPR interference, which is really a dead Cas9 that's conjugated to CRAB, which is a transcriptional repressor, and designed guide RNAs that tile across this exon -3 region, thinking that if we're able to knock down expression of exon -3, perhaps that affects FOXR2 expression. And indeed, that seems to be the case. There's actually one guide, this fourth guide, the most distal guide, that is unable to knock down FOXR2 expression, but all of the other guides are able to, suggesting that indeed this promoter is required for FOXR2 expression. This is just quantified below. And actually, more recently, we've taken the specific exon -3 sequence and shown that it's sufficient to activate luciferase in a luciferase promoter assay, which you can see here. So the exon -3 promoter sequence is sufficient, and then we've been trying to narrow down the sequence to a minimal promoter region that's sufficient to activate luciferase. We also are really interested in examining the epigenetic landscape, given we didn't find SNVs or copy number changes in the non-structural variant tumors. And to do this, we collaborated with a group at the Broad Institute, especially David Wu and James McFarland, who have been really critical for this work in analyzing reduced representation by sulfite sequencing to look at methylation status. So what you're seeing here is across all of the cancer cell line encyclopedia cell lines, all these are in gray, indicate that they don't have FOXR2 expression, and there's about 50 cell lines that actually do have FOXR2 expression. Across the x-axis, what you're seeing is a region of the X chromosome, and each of these columns is actually a different CPG site. And what we found actually is that within FOXR2 expressing cell lines, there's clearly a region that is hypomethylated, which is indicated in blue, relative to non-FOXR2 expressing cell lines, which was very striking. We see this shown just in a different way, but if you look at the correlation between methylation at each CPG locus and FOXR2 expression within this specific FOXR2 region, this is very left shifted, indicating that across that specific locus, they're much more likely to be hypomethylated compared to any other cell line that doesn't express FOXR2. What is also interesting is if you look at normal methylation data, so this is ME-DIPSEQ data from UCSF in fetal brain and germinal matrix, normally this region is actually methylated. And we reason that this may be an instance of something called a cancer testis antigen, which has been documented in the literature, which suggests that normally these cancer testis antigens are actually methylated and undergo some demethylation process whereby an oncogene is activated. So we think this is likely the mechanism by which FOXR2 is activated in the non-structural variant cancers. I'm gonna skip this for now. Going through some of our functional data, we did this a couple ways by looking at the dependency map data from the Broad Institute. So on the left, this is across about 900 cancer cell lines that underwent CRISPR-Cas9 knockout screens. And then on the right are a conglomeration of a number of different RNAi knockdown screens. And what you see here is that both for CRISPR-Cas9 knockout and RNAi against FOXR2, for FOXR2 expressing cancer cell lines, knockout or knockdown of FOXR2 is actually the #1 dependency, suggesting that FOXR2 is indeed required for proliferation, at least in vitro. We've also done these experiments actually in low throughput using the CRISPR-I guides I mentioned before, but also some more classical CRISPR knockout guides against exon 1. And we've done this in a number of lineages because we wanted to sort of show the relevance across all human cancers. So we've done this in DIPG cell lines, in non-small cell lung cancer and melanoma cell lines, all of which utilize the exon -3 mechanism of activating FOXR2. And you can see in all of these instances, knocking down FOXR2 attenuates proliferation. We've also tried to show sufficiency in a number of contexts, and that includes in vitro. So we've done work in H9 human neural stem cells, mouse neural stem cells, as well as these AALE cells, which are transformed lung tracheobronchial epithelial cells. And these are all isogenic models in which I introduce FOXR2 relative to a vector control and then measure confluence, or in the case of mouse neural stem cells, they grow as neurospheres, so spheroid size. And you can see in all instances, FOXR2 accelerates proliferation. We've also really recently done this in some melanocytes as well and see something similar. We had also made this sort of interesting observation that at least in FOXR2 expressing brain tumors, that tumor locations seem to correlate with the type of mechanism of activation. So cortical gliomas were much more likely to have these enhancer hijacking mechanisms that utilized a fusion or structural variant, whereas the midline gliomas, including DIPGs, were far more likely to use these alternative exon mechanisms. And so we wanted to be able to create models of these different regions of the brain. And to do that, we harnessed an in vivo mouse model using in utero electroporation. This is all work that's been done very closely with Tim Phoenix's lab, who's at the University of Cincinnati, who has really pioneered a lot of the in utero electroporation specifically for brain tumor models. So this is a cortical glioma model. So essentially your plasmid of interest is injected in utero, and this results in spontaneous glioma formation. And so we're able to generate Kaplan-Meier curves. We're able to take bright field images of the tumors, but we're also able to take sections at the end and review them. So you can see that FOXR2 in blue rapidly accelerates mortality in these mice. These are very, very bulky tumors that are also labeled by GFP. And then we've actually had all of our sections analyzed by one of our neuropathologists here at Dana-Farber and at the Brigham. And they've confirmed that these do look like gliomas. And we've also looked at a number of glial markers as well as Ki67. You can see there's clearly proliferation in the FOXR2 expressing tumors. We've done something very, very similar in brainstem gliomas. This is a little more technically challenging, but we're able to generate brainstem DIPG models, and we see something very, very similar to the corticals, where FOXR2 really accelerates growth of these tumors. You can see they're very, very bulky in these images, and again, increases proliferation through Ki67 staining. We've also looked at some glial markers just to prove to ourselves that the glial markers are still there. And indeed, you see, at least for OLIG2 and GFAP shown there, that we still have good staining. And again, as a sanity check, that FOXR2 expression is really only in FOXR2 expressing tumors, which you see nicely here. So this data really shows that FOXR2 is certainly necessary in FOXR2 expressing cancers for proliferation. We've seen that through the dependency map data, as well as some of our low throughput assays. And then we've done quite a lot of work looking at sufficiency in both in vitro and in vivo models to show that it's a very potent oncogene. I'm not showing you data here, but we've actually also done some subcutaneous flank injections in which FOXR2 by itself is actually sufficient for flank tumor formation. So the last part I'm going to talk to you about is the contributions of these different FOXR2 domains and really trying to tease apart the mechanism by which it mediates oncogenesis. So it's actually known that FOXR2 has a MYC binding domain here, and then it has its sort of canonical fork-head DNA binding domain, which is what makes it a FOX protein. So we designed constructs in which these domains were deleted, and then introduce them into our in vitro models as well as our in vivo models to see what would happen. What we saw was actually that deletion of the MYC binding domain seen in red attenuates proliferation, but also deletion of the fork-head DNA binding domain actually attenuates proliferation as well. They both grow a little bit faster than the controls, but they certainly attenuate proliferation relative to wild-type FOXR2. And we actually see something very, very similar in the in vivo context. If you look at the percent of Ki67 positive over GFP positive cells that indicate that the transfection has occurred. So this was sort of surprising to us. There's a lot of literature on FOXR2 and its ability to bind MYC and stabilize MYC and form a complex, but there's been much, much less focus on sort of the downstream fork-head related effects of FOXR2. So to try to explore this a little bit more, this is also just showing that we've done a lot of cyclohexamide assays to inhibit translation to show that indeed this MYC binding domain is really important. So these are just cells expressing FOXR2 versus cells expressing control. They're exposed to cyclohexamide for up to 5 hours. And you can see that in the control, MYC is actually degraded very quickly, but in FOXR2 expressing cells, we actually maintain MYC expression really nicely, even after 5 hours of cyclohexamide exposure. So to get at this question of the fork-head domain and really where does FOXR2 bind, we actually did, we had a tagged version of FOXR2 that was an HA FOXR2 introduced into one of our isogenic human neural stem cell models. And then we performed HA chip-seq to really try to figure out where FOXR2 is binding. And when you do a motif analysis, once you generate the chip-seq data, what was interesting to us was, well, we expected to see MYC, which are coming up as motifs here, which is sort of not unexpected, but we were surprised to see these other transcription factors popping up as some of the top motifs where FOXR2 is binding. And ELK1, ELK4, ETS, these are all members of the ETS transcription factor family. And this was very surprising to us. And we also found that FOXR2 seemed to bind preferentially to these sites. There's 29 ETS transcription factors compared to 29 random transcription factors. So this was really surprising that FOXR2 had some relationship with this other ETS transcription factor family. And what we've done since then is actually a very large amount of transcriptomics, both in human cancers across a number of different lineages, including osteosarcoma, different types of gliomas, melanoma, lung cancers. And then we've also used our in vivo mouse models and additionally have used our cancer cell lines. And we've also used our cancer cell lines and knocked down FOXR2. And what we see when we run gene set enrichment analysis, when you knock down FOXR2, you knock down FOXR2 target gene sets. That's that's good. That's a good sanity check. But almost all of these gene sets are actually related to ETS transcription factors. And in the context of FOXR2 overexpression, you see something very similar, but in the opposite direction. FOXR2 target gene sets are up, but all of these ETS transcription factor family gene sets are highly upregulated. So we're really currently trying to really understand the relationship between FORCAD transcription factors, ETS transcription factors, and how specifically they interact, whether that's at the protein-protein level, protein DNA level, or even protein RNA level. So this is ongoing work in our lab. I also, just as a physician scientist, wanted to point out that and briefly highlight some work being done by our collaborators in the Department of Chemical Biology at Dana-Farber in Eric Fisher's lab. This has been led by a postdoc, Sarah Dixon-Clark. And this is in its early stages, but we've been working really closely with them to try to target FOXR2 using actually a degradation strategy, where you can exploit the E3 ubiquitin ligase system and basically trick it into degrading your protein of interest, which in this case is FOXR2. So we've sort of Sarah has really nicely shown some data using this FOXR2 reporter that she has, and is currently working on a CRISPR E3 library screen to identify and validate potential E3 ubiquitin ligases that can be used to really degrade FOXR2. This is in its really early stages, but our hope is that this work has some translational impact, not only for pediatric brain tumor patients, which we're personally interested in, but actually across adult and pediatric cancers. So I've shown you that FOXR2 is a very potent cancer oncogene across human cancers. And my hope in my future research career is really to continue dissecting how oncogenes are activated through both these genetic and epigenetic mechanisms. And I just want to highlight, this has been a tremendous collaboration across a number of groups. Thank my postdoc mentor, Mimi Bandopattai, who has been just tremendous, who is also a physician scientist and has been a really great example of what that looks like. And I also wanted to highlight a very talented research assistant in our lab, Dale Wang, who has helped me with a lot of these experiments, who also will be hopefully a future physician scientist in a few years. And then Tim Phoenix at the University of Cincinnati. And I just want to highlight the group in Heidelberg and the INFORM Consortium, who's been pivotal for the German tumor samples. And then we have some collaborations at McGill and of course at the Broad Institute. And then funding sources just from St. Baldrick's, Alex's Lemonade Stand, and Helen Gurley Brown Foundation, as well as Griffin's Guardians. So I'm happy to take any questions.
Thank you so much, Jess. That was a really good talk. We do have some questions. I had one to start us off. Is it known what the cell type of origin is for the DIPGs? Is it, because I saw some oligo genes expressed, and you had looked at some astrocyte genes in terms of proliferation. Is this known?
Yeah, it's thought to be a very early glial precursor, although there are a number of groups that are actively doing single cell on the actual patient samples. There was a paper published a couple years ago that only looked at, I think, 4 or 5 samples, but there's ongoing work in our group and some collaborators looking at this. There's also actually a really cool single cell data set from some other pediatric brain tumors looking at cell of origin across all pediatric brain cancers. But this is something we're pretty interested in, especially for FOXR2.
Okay, so one of our questions from the Q&A list is from Basilika Tasik, and she wants to know, what's the mechanism for focal amplification? How many copies of the MYC enhancer occur in amplified cases?
So for that particular instance, it's actually really interesting. So some of them are, you can see that it's a very simple focal amplification where there might just be 2 or 3 additional copies. But there are others, including one that I'm thinking about, where it's a very, very complex structural rearrangement that actually involves much more than just that specific enhancer site, but almost crossing all the way over to where MYC is located on chromosome 8. But what's interesting is that they all share this consensus sequence that lines up with the enhancer. So if you look at the preprint, what's also interesting is that we think that some of these structural variants are actually dictated by some of the earlier genetic events that happen in the tumors. And you can see that there are actually a number of different signatures that come out in the pediatric hypergliomas when you look at the structural variant landscape. So there's a lot of work that people are doing right now to try to suss that out a little bit more. But to answer your question, it's highly variable. Some of them are very simple and some of them are extremely, extremely complex rearrangements.
Okay, our next question is from Trygve Bakken. He says, hi Jess, do you have any attack seq data from FOXR2 positive tumors? Any evidence for enhancer activity linked to the novel promoter?
I literally just got an email that I think our data might be ready. So more to come, but nice. We're pretty interested in that. And I'll hopefully have an answer for you soon. I can send you a note when we know.
Great. Here's a question from YouTube. I'd like to ask Dr. Tsai if there is a possible correlation between FOXR2 activation and the codon usage bias at the mRNA level.
Huh. I haven't thought about that actually. That's an interesting point. I'm not sure anything is known about that in relation specifically to FOXR2, but it's definitely something worth looking into. But I don't know the answer to that question.
Okay. Ben Slivka wants to know, if you upregulate FOXR2 in a non-FOXR2 cancer, does it increase proliferation in that cancer?
So it does. It does increase proliferation, and we are working on trying to figure out if there are dose-related effects to. So you can imagine that you have a cancer that expresses MYC already, right? And so you add FOXR2 and accelerates proliferation. Is there some dosage of MYC where you're stabilizing MYC to a certain extent where that level of MYC expression actually becomes toxic to the cell? So it seems like there's actually almost like a sweet spot for the amount of MYC and the level of MYC that a cell can tolerate at any given time. We're also trying to do something similar, but we don't have the data yet, where we add FOXR2 in an inducible way, right? So where you can turn on and off FOXR2 expression and see what happens in terms of differentiation and cell state. So those are all things that we're working on.
Okay. Mike Lodato says, great talk, Jess. Given that FOXR2 increased expression and MYC mutation seems mutually exclusive, do you think FOXR2 is transcriptionally regulating the MYC locus itself?
It's a good question. I don't think we have good evidence that it's regulating the actual locus, only that they physically bind together. Although we are currently in the process of generating additional data. I'm trying to see if I can actually endogenously pull down FOXR2 in some cancer cell lines. But I think all we know now is that there's a physical interaction between FOXR2 and MYC, not necessarily that FOXR2 is binding at the MYC locus, or even MYC-N or other MYC isoforms. But more to come on that.
Okay. Just a couple more questions. Lauren wants to know a more general question. How would the data at Allen Institute help for more translational research like you do? A lot of brain science work is on healthy tissue. So just kind of wondering how you might use tools that exist.
I think cell typing is really critical for us, especially as we're going into doing more single cell of some of these precious samples and being able to identify cell of origin. I actually also think that normal expression across development is something that is really, really helpful for us. And we've looked in the Allen set to try to see if there's FOXR2 expression in any of the data sets there, and we can't find it. So those data sets are actually tremendously helpful, especially for cancer biology, and then across development, especially for people who are interested in pediatrics and development, which a lot of people, think that pediatric cancers are really a problem of development, right?
Yeah. Final question to answer live. There probably will be some questions you can follow up on in the Q&A, but Shinbo Lee wants to know, is there any mutation in FOXR2 in cancer? What is the half-life of FOXR2?
I don't know what the half-life of FOXR2 is. That's a very good question. We should find that out. There aren't any known S and V mutations that I know of in FOXR2, and we haven't found any in our analysis either. But we're trying to do We're trying to do a couple other types of sequencing. So we actually have long read sequencing cooking for a couple of tumors to try to better assess this question and make sure we're not missing anything. But there is no FOXR2 mutation that I know of, at least now.
Okay, thanks, Jess. That was great. We're going to take a 10-minute break before our next presenter. So we'll see you all back at 10:50 Pacific time to hear from our next NGL speaker. Thank you.
All right, welcome back everybody from the break. My name is Saskia DeVries. I'm an associate investigator in the MindScope program, and I'm going to be the MC for this last session of our showcase. So our next speaker is our next gen leader, Lucas Pinto. He's an assistant professor in the Department of Neuroscience at Northwestern University. His talk today is titled Large-Scale Mechanisms of Flexible Decision-Making. So to study flexible decision-making, he uses navigational decisions in virtual reality using optical recording and perturbation tools. He's presenting his work demonstrating that more demanding tasks engage more distributed circuits. So welcome, Lucas.
Thank you so much, Saskia, for the introduction. It's really a pleasure and an honor to be here. And it's been super cool to hear about the amazing research being done both at the Allen and also by my fellow NGLs. Before I start, I would like to acknowledge that I do work and I live on the traditional homelands of the people of the Council of the Three Fires here in modern day Chicago. And I would also like to thank Cindy Poo for her acknowledgement of the issues with the Brazilian natives, being myself a Brazilian American. My lab is very interested in understanding both how local circuits perform cognitive computations, but also how they flexibly interact at large scales to enable different kinds of cognitive behavior with a particular focus on decision-making. And in particular, we're very fascinated by our ability to really adapt the way we make decisions to meet ever-changing demands for the environment in a way that is very crucial for survival. For example, if you're crossing a busy intersection in Chicago, you need to decide to look to the left first, but a similarly busy intersection in London requires you to first look to the right. And if you're an American tourist in London in this case, and perhaps you zoned out because you were looking at Twitter on your phone, what this required you to do is realize that you're in a different context, perhaps by realizing that there are red double-deckers everywhere, and then in turn retrieve a different decision rule from memory and apply that decision rule accordingly. So these decisions are sort of similar in terms of their overall perceptual difficulty, but they differ in terms of their basic rule. Decisions can also vary in terms of their perceptual difficulty. So consider this other very simple example. Say you're driving in traffic on a clear day and you encounter a red traffic light. It is pretty easy to decide when to step on the brake, but on a stormy night, it becomes a lot harder to distinguish between the rear lights in the cars and the red traffic lights, such that you might need to stare a bit longer through your water splashed windshield before deciding to step on the brakes. So really what I'm trying to say more generally is that decisions can have different underlying cognitive computations. So on a clear day, low-level sensory processing of the red traffic light, for example, should be enough to trigger the appropriate action of stepping on the brakes. But on a stormy night, you might need to add these intermediate time-extended computations to accumulate the noisy sensory evidence coming in through your windshield. You also may or may not be required to hold something in memory before you actually commit to an action. And much like crossing the street in London, we're often required to detect changes in our overall behavioral context and in turn readapt and reorganize our behavior accordingly. So in my lab, we're interested in understanding the neuromechanisms behind all of these different aspects of flexible decision-making. But for the better part of my talk today, I will focus on work that I did as a postdoc with David Tank and Carlos Brody at Princeton University, where we asked what are the differences in large-scale dynamics between decisions that vary in terms of their underlying cognitive demands. And in particular, like Saskia mentioned, we went in going with this hypothesis that more complex demanding decisions really require the recruitment of much more widespread cortical regions. So in order to actually tackle these questions, what we had to do was to develop different decision-making tasks where we, to the extent possible, held constant the low-level properties of the stimuli that the animals experience, as well as the actions that they took, and vary everything that happened in between these sort of sensory and motor aspects of the task. A very convenient way of accomplishing this and a very powerful and well-controlled way of doing behavior in general is using virtual reality for navigating mice. We essentially train mice to play these very immersive video games, and their joystick is this styrofoam ball that is compressed air, and that allows them to run with very little friction. And in turn, we can read using optical velocity sensors displacements on the surface of this ball and transform those into translations and rotations of a virtual environment that we project in closed loop onto a screen that covers most of the field of view of the animals. And an additional advantage of these virtual reality systems is that the animals are head fixed, which means that we can combine a number of different optical tools to either record or perturb neural activity with different kinds of spatial temporal resolution. So just to give you a sense of what this actually looks like, here is a video of a mouse navigating a virtual environment. In this case, he is navigating a virtual T-maze. What he's trying to do is find where the reward is, either on the left or on the right side of the maze at the end. Notice, for example, how well this guy can control the ball, even though he is running at speeds that literally exceed 0.5 meter per second, or about 2 feet per second. You can also see here this device that we built to perturb cortical activity in these mice, as I'll get to in a second. So we developed several tasks, and in the interest of time, I'm going to focus on 2 of such tasks. The first task that we developed was a demanding task for the mice that required them to accumulate sensory evidence, and as well as hold information in short-term memory. We call this the accumulating towers task. So we trained the animals to navigate down the virtual T-maze that you saw in the video in the previous slide. This is a maze that is 3 meters in length, which takes them about 6 or 7 seconds to run through. And as they do so, during the first 2 meters of the maze, they encountered these tall white objects, which we call towers, and these appear transiently on either side of the maze. And after a delay of 1 meter, their job is to turn to the side where they saw the highest number of towers in order to retrieve a fluid reward. Importantly, the towers, the counts of the towers, the side with the most towers, and the exact position of the towers vary randomly from trial to trial, which one gives us a whole range of trial difficulties that we can leverage for behavioral analyses, but importantly means that the animals cannot solve this task by simply memorizing positions of the towers, for example. And indeed, we can use behavioral modeling to show that the animals solve this task by taking into account evidence that comes from multiple parts from throughout the maze before they make their final decision to turn to the left or to the right. So this is a demanding task that we developed. A much easier task for them is the visually guided task. This task happens in exactly the same maze with exactly the same tower stimuli. But additionally, from the very beginning of the maze, they can see this tall visual guide at the end of the maze, which tells them where the reward is. And this completely removes the need for evidence accumulation or short-term memory. And indeed, we can show that the mice perform this task at much higher levels, suggesting that it is in fact easier for them and that they do not count towers in order to solve this task. So now that we have these tasks at hand, we can ask what are the differences in large-scale dynamics between these 2 different kinds of decision making. And in particular, the first question that we asked was what are the regions of the dorsal cortex that are required for the mice to perform either of these tasks. And for this, we used the approach pioneered by Carl Svoboda's lab, where we rendered the intact skull of the animals optically transparent, and we used the scanning laser system to randomly target different cortical regions through the transparent skull. And we did this in animals expressing chiral-rhodopsin in inhibitory interneurons in the cortex, such that when we shine a blue laser with our system, we activate these interneurons, which in turn effectively silences the activity of a local patch of cortex. We then divided up the dorsal cortex, going from sort of premotor areas in the front all the way to the visual cortex in the back, into a grid of 29 locations, which we targeted for this first experiment bilaterally and throughout the entire duration of randomly interspersed trials in behavioral sessions in which the animals performed one of the 2 tasks. So what did we find? First, let's look at what happens when we do these experiments as the animals perform the easier visually guided task. What you're looking at here is a map of cortical inactivation effects using circle representation such that the center of the circle indicates the location of the laser beam. The diameter of the circle tells you the size of the effect, in this case, how much performance drops when that certain cortical area is inactivated. And the color of the circle gives you the statistical significance of the effect, such that wide circles indicate non-significant effects, and progressively redder colors will indicate lower p-values. So here, quite clearly, when they're performing the visually guided task, what we observe is that the effects are very localized and in fact restricted to areas in or around the visual cortex of these mice, and to a much lesser extent, this medial premotor area here. Really, this is very compatible with other findings in the literature done sort of outside of this virtual reality context and also with other sensory modalities. And so there were no surprises there. However, when we performed these very same experiments as the animals performed the accumulating towers task, the more demanding accumulating towers task, we observed a vastly different pattern of inactivation effects, and this time widespread effects of inactivation throughout the cortex. For this particular laser power that I'm showing here, which is exactly the same laser power as we used for the visually guided task, we actually observed significant effects of inactivation in every single location that we tested. Now, we do think that this is probably somewhat of an overestimate given the spreads of inactivation that we measured in the cortex. But we did perform lots of control experiments with different laser powers and in animals not expressing chernodopsin, and really convinced ourselves that the animals do require a lot more cortex to perform this more demanding accumulating towers task. Now, the question, of course, is what are the actual contributions of different cortical areas? Or to begin with, are they different at all? Already from this data set, we knew by analyzing other behavioral indicators, for example, the side biases that the animals displayed and sort of motor aspects of their performance, that really the effects of inactivating different cortical areas differ in nature according to the cortical area. But to answer this question more directly, what we then did was to select a subset of 7 cortical locations, which we again targeted bilaterally, but this time during much briefer epochs throughout the behavioral trials. And really what this allowed us to do is to look with a bit more granularity into the computational contributions of different cortical areas. And for example, it allowed us to ask what happens to the way the animals use sensory evidence that is occurring while the laser is on, or either before the laser came on or after the laser is off. And indeed, we saw some really interesting differences between the effects of inactivating different cortical regions when we looked at these things specifically. For example, let me walk you through what happened when we inactivated the posterior parietal cortex, or PPC for short, in the ways the animals used evidence occurring during the laser or even before the laser came on, that is the memory of the evidence. So in particular, what I'm showing you here are summaries of a logistic regression model that we developed, where we try to explain the final choice that the animal makes based on the sensory evidence that they experience in different time points in the trial, aligned by when the laser came on, or even, or in this case, when the laser came off. So specifically what I'm showing on the x-axis is these different time points with respect to either laser offset or onset. And on the y-axis, I'm showing you a normalized coefficient that indicates the effects of the laser on evidence use, such that 0 would indicate that the laser does not affect the way the animals use sensory evidence. And negative 100 means that the animals completely stop using sensory evidence from that time point in the trial. So while the laser is on, we observe a very dramatic decrease in the use of sensory evidence when we inactivated PPC. And here we're combining all epochs together as a summary. And this perhaps indicates a role in more immediate sensory processing of the stimuli. But perhaps even more interestingly, we observe a near complete drop in the use of sensory evidence for evidence that occurs immediately before the laser came on. So in other ways, very recent evidence. And this effect really decays as you go further in the past. And basically what this tells us is that the PPC appears to contribute to evidence accumulation on short timescales. And we observed somewhat similar patterns across posterior areas of the cortex, but we observed something different when we inactivated more frontal areas, for example, this medial part of the premotor cortex. Here, we observed much less of an effect while the laser is on, indicating perhaps not such a big role in immediate sensory processing, but a much longer deficit in the use of sensory evidence going much further into the past, suggesting that this area contributes to this behavior on longer time scales. So to summarize what I've told you so far, when we performed sort of systematic inactivations of the dorsal cortex in a simple task, we observed very localized effects of inactivation, but these became much more widespread when the animals were required to accumulate evidence over seconds, such that the effects are compatible with this idea that frontal areas contribute to the behavior on longer time scales than posterior cortical areas. This, of course, begs the question of what are the differences in the large-scale dynamics across the cortex that explain these really dramatic differences in the effects of cortical inactivations. And so to answer this question directly, we again took advantage of this transparent skull preparation, but this time applied to animals expressing the calcium indicator GKIM6F in cortical excitatory neurons. And then in order to be able to image from the whole dorsal cortex simultaneously, we used the wide field macroscope with mesoscale spatial resolutions of about 50 microns per pixel. Importantly, we did these experiments as the animals switch between these two tasks within the same behavioral session, which really gives us a nice paired experimental design. And in fact, the movie that I'm about to show you is an example movie from one of these behavioral sessions coming from a single mouse performing this task switching paradigm. And even though it's an example, I think it really nicely illustrates a lot of the main findings from these experiments. So in these movies, on the left here, I'm going to show you activity that is normalized and pseudocolored for each pixel. So when a pixel appears yellow, it has higher activity. And this is going to be averaged over every time the animal correctly turned to the left side of the maze. And here on the right, you will see an example trial progression just to orient you with respect to the timing of the activity. And before I even start the movie for the visually guided task, I'd like to point out that as soon as the trial starts and the animal sees this left visual guide at the end of the maze, we do observe a brief activation of the contralateral visual cortex, as you might expect, and this is a nice sanity check. But after that, really not much happens. The cortex is sort of like not doing anything super meaningful as far as we can tell until the animals gets to the end of the maze and we see this large increase, a correlated increase in activity across the cortex. For a number of reasons, we think this is some sort of contextual signal related, for example, to the turning or reward expectation, or maybe even both. In fact, in the accumulating tower task, we see this correlated increase in activity at the end of the maze as well. But additionally, much earlier in the trial, we see very widespread dynamics across the entire dorsal cortex. And this happens including as the animals are accumulating evidence. It's maybe easier to see with the still frame here and compare with the other task in which, remember, they are still experiencing the towers. We, of course, wanted to know if this earlier widespread activity is in fact related to task or decision-making computations. I won't really have time to go into this in detail, but suffice it to say that we devised a bunch of different decoding and encoding models of neural activity. For example, we used different behavioral events such as the occurrence of the towers and the running tally of tower counts and turning and speed and so forth to sort of try to explain variance in the activity of these different cortical areas. And we indeed found that activity can be explained by different decision-making computations across the dorsal cortex, really in a way that is much more quantitatively rather than qualitatively different across different cortical areas. In other words, task information is widespread across the dorsal cortex. We did, however, observe a very interesting difference across different cortical areas because in addition to using behavioral indicators in our encoding models, we also used autoregressive coefficients across different time lags, which you can simply think as the sort of autocorrelation function of different cortical areas. What I'm showing you here are exponential decay fits to these coefficients. for the different cortical areas. And as you can see, the autocorrelation of activity decays really quickly for posterior cortical areas, for example, V1, but do so much slower for frontal areas such as the premotor cortex. This is even easier seen in the time constants that we extracted from these exponential fits, showing a very systematic increase in the intrinsic time scale of activity going from visual areas all the way to frontal areas in the cortex. And of course, this in of itself is not novel. Lots of groups before us have very beautifully demonstrated the existence of this intrinsic time scale in the cortex, but perhaps what this is showing is that this time scale is still at play during cognitive behavior and perhaps is co-opted as a mechanism to accumulate evidence on distinct time scales depending on the cortical area, which really goes well with our inactivation findings that I showed you a bit earlier. So to conclude this sort of data part of my talk, I hope to have convinced you that different decision-making tasks are associated with distinct dynamics across the cortex, such that more complex tasks are associated with more widespread and complex decorrelated activity. And that perhaps within a task, parsimonious principles such as intrinsic timescales could explain the logic whereby different areas are recruited. And so just to wrap up, I would like to give you a quick sneak peek of how we're following up on these results and ideas in my own lab. But before I do so, I would just like to give a quick shout out to big team science and point out that really this work was done as a part of a much larger collaboration funded by the BRAIN Initiative across 7 different labs at Princeton University and at UC Davis, where this whole fantastic team uses the same accumulating tower SAS that I was telling you about today to study the role of different circuits throughout the brain in working memory and decision making. So now with just a couple of minutes that I have left, I will tell you a little bit about what we're interested in my own lab at Northwestern, and also what we've been up to since we opened doors a little less than a year ago. So I would say broadly, we're interested in 2 sets of questions. The first is what are the actual circuit mechanisms that control task-specific cortical dynamics and also our ability to seamlessly switch between different decision-making tasks. And also, how do different cognitive computations determine the spatial temporal features of these rich dynamics that we and others observe? Or to put it another way, what about a task makes the cortex act the way it does? And so just to make things a little bit more concrete, one thing that we're very interested in is on the neuromodulatory mechanisms of task-specific cortical dynamics. For example, my amazing postdoc, Amy Rapp, has been leading this project to understand the role of cholinergic projections throughout the cortex in sort of controlling cortical dynamics in a task-specific fashion, with a particular emphasis on the heterogeneity of these projections, which is becoming increasingly appreciated. Just to illustrate this, let me show you this very cool analysis that Ryan Liu, who was a rotation student in my lab earlier this year, did using this amazing resource that the Allen has for the community, which is the Mass Connectivity Atlas. So what Ryan did was to select anterograde tracer injections targeted at cholinergic neurons from across different subnuclei of the basal forebrain and look at their patterns of innervation of different cortical areas. And this is just a sort of max projection as well as some quantification of that of those patterns, showing this really beautiful, strong segregation of basal forebrain inputs to either posterior cortex or frontal cortical areas. Another project that I'm super excited about is being led by my very talented graduate student, Peter Salvino. Peter has been developing a new decision-making task that will really allow him to sort of differentiate between different underlying decision-making computations, particularly looking at how varying their difficulty and their duration impacts cortical dynamics, both at the level of whole cortical areas, but also at the level of cortical microcircuits, with a particular eye on the role of intrinsic timescales. And so with that, I would just like to thank the people who've made this possible. in particular my postdoc advisors at Princeton, Carlos Brody and David Tank, as well as lots of collaborators with a highlight for a few key collaborators. Shout out to Kanaka, my friend and collaborator, who you'll have the pleasure of hearing from shortly. And also like to thank my amazing team that I've managed to recruit to my new lab at Northwestern, as well as my funding sources, the Brain Initiative and the Simons Foundation. And thank you so much for your attention. And hopefully I still have time for questions. Thank you.
Great, thank you so much, Lucas. That was a wonderful talk. I will dive right into questions. And if anybody has any, please add them to the Q&A and we'll get to as many as we have time for now. So Casey has a question, which is, if you inactivate cortical areas on one side only instead of bilaterally, are all areas similarly robust in the accumulating towers task?
So we only did preliminary experiments. That's a great question. Yes, it appears to be the case. I mean, there obviously the pattern of effect varies. Instead of having sort of just like overall decreases in performance, what you get is side biases in the directions that you would expect. I don't have quite enough data from that to be completely confident in my answer, but that's what the preliminary data points to.
Great. Another question here, have the same animals been used for both tasks?
So not for the optogenetic experiments, because the way we design those is that we prefer hitting every single area within the same behavioral session. And at the same time, we cannot hit these areas too often because the animals just get generally unmotivated and stop performing altogether. So what this means is that we only have a very limited budget of trials where we can inactivate. So the way we designed those experiments was to just do one behavioral task per animal. Now for the imaging experiments, the same animal is in fact switching between the two different tasks within the same behavioral session. And so, for example, that movie that I showed is coming from the same animal on exactly the same day and the same sort of the same session. So those differences are consistent across animals.
And so do you think it's possible, so are they doing the same task in the same day?
Yes, they're doing the two tasks on the same day, yeah.
And so there's kind of a follow-up question here is, could there be biasing of activation resulting from the previous tasks or trials in when you're switching between the tasks like that?
So that's an interesting question. We haven't directly looked into this. So just to clarify, sort of like the inactivation experiments are done in separate animals, right? But as far as we can tell, there aren't super strong trial history effects related to either the tasks that the animal had been performing or other kinds of trial histories. But I will say that I haven't looked at this carefully enough. So there could be interesting things there.
Interesting. So I'll just ask one more little question. So you, in the regression model that you showed for the two different, the regression model you showed, and you're comparing two different areas and kind of how much it affected how the effects on the regression model in these two different areas. Was that model performance kind of similar in those areas? Was it explaining roughly the same amounts, or do you see kind of different? Yeah.
That's a great question. It explains almost it's completely statistically indistinguishable. It explains choices just as well in sort of regardless of the area that we're inactivating, actually, which is a very important question to ask for sure. But yes, we're sort of confident that those effects aren't coming. And we also did lots of sort of like shuffling controls. So, and these are very important because there is variability across animals, and not every animal experiences every single condition, even though there's overlap. And so, yes, we did make sure that it's not just like random variance coming from animal variability or experiment variability. Yeah. We really believe those results. Great question.
Right. And then I see there's a question that popped up in the chat. Is there a mouse model for frontal temporal dementia, perhaps useful for improving human performance?
So I am embarrassed to say that I know very little about sort of models of, I know that there are models of Alzheimer's in mice, which is also relevant and sort of pertains to sort of all sorts of questions that we're asking. I'm not sure about frontotemporal dementia, but yes, I think that those are, it's an interesting question, and it could be, paradigms like ours could be relevant for studying like these, pushing the sort of cognitive behavior on the mouse end, I think could lead to insights for these sort of disorders.
Excellent. If anybody has any more questions, add them to the Q&A, and Lucas will be able to go in and answer those later on. But thank you again for that great talk. That was really excellent and exciting work. Great, so our next, we have a team talk from our genomics team called Defining Cell Types in Health and Disease by Single Cell Genomics in Mouse and Human. Our cell types program has been working on profiling cells from adult humans and mice by their transcriptomic profiling. And they're going to discuss some of the next phases of this work, which includes epigenetic and spatially resolved data in the adult mouse, aging in the healthy mouse brain, and pathology by cell type in Alzheimer's. So welcome to the genomics team.
Hi, everyone, and I'm Zizhen Yao, Assistant Investigator from Informatics and Data Science Department. We have a very exciting team talk today on defining cell types in health and disease by single-cell genomics in mouse and human. So we have team members from both mouse and the human genomic team telling you the latest progress we've made. So our session includes 4 short talks. I will start with the introduction, giving the high-level overview of what we've done on building the whole brain cell type atlas for adult mouse and other ongoing projects. Amanda Mitchell will tell you about the Multiome platform development, which is essential to identify cell type specific enhancers. And Kelly Jane will tell you the progress we made in categorizing cell types in the aging mouse. Rebecca Hodge will tell you about genomic applications for understanding human disease. So the mission of the cell type program at Allen Institute is to build a part list of the brain and to provide multimodal categorization of each component. And the single cell genomics are at the heart of these tremendous endeavors. So over the years, we have established 2 highly efficient and robust single cell profiling platforms, SmartSeq and 10x. SmartSeq is low throughput but high quality and provide full length coverage, while 10x Chromium is low throughput, but is high throughput, but have relatively low gene detections that only profile the 3 pairs of the transcripts. So we profile the cells in both platform and leverage their strength and build integrative transcriptome taxonomy. So once the taxonomy is built, it will be used as the anchor point and to link with other data modality, including mFISH, morphology, connectivity, analogy, and cell type specific markers will be used as candidates for genetic manipulations for building genetic tools. So for the mouse whole brain atlas, we actually finished the data collections already altogether. So with a total of 5.2 million cells collected using both SmartSeq platforms and 10x version 2 and a version 3 platform. So we have a close to 2 million cells in isocortex, and the other 3 million spread among the various cortical areas. Now we have a first draft of the high resolutions cell type taxonomy for the whole brain using 10x version 3 data only. And this is built based on the young adult around age 56 and cover both sexes. So what I show here is a 2-dimensional is a low-dimensional embedding of the reflecting all the cell type diversities using UMAP. So I have a UMAP here colored by class, by regions, and by subclass. So in this UMAP, you see the non-neuronal populations on the top, and then the GABAergic types and the glutamatergic types. And then also identified glycinergic and dopaminergic and other type of neurons that allow us to link it with literature. So in this original map, you see pretty extensive original gradient that for both GABAergic and glutamatergic neurons. In both cases, you see the transitions from cortex to cerebral nucleus to hypothalamus, midbrain, pons, and hindbrain. And you see similar gradients in the other directions. So we try to reduce the overall complexity of this taxonomy by grouping cells in subclass, but unfortunately, due to all various types of gradients, it's a very challenging task in these subcortical areas, midbrain and hindbrain. So having the first draft of this, the whole brain cell type atlas in DAO has a huge impact. First, it provides a blueprint of almost all the cell types from the mammalian brain. It provides critical tools to interpret any gene or genetic tool specificity in context of the whole brain. It can serve as a Rosetta Stone for multimodal data collections, and then allow us to choose cell types for this study more strategically. And it can also serve as a starting point for evaluation of development, aging, disease, and cell states and plasticity. And finally, it helps us to interpret cell types and assigning putative functions in other species by aligning to this mouse reference cell type atlas. So we have been doing that very successful in cortical areas. Now we hope to scale it up for the whole brain. So next, I give you some really summary of all the ongoing projects that we have either actively in progress or in planned. Boasting has been working actively to build adult cell type taxonomy. And so for a mouse, we have finished data collections on whole brain, spinal cord, and DRG, and the next in line. For human, the effort is going to focus on basal ganglia, thalamus, and midbrain. So we're now extending our efforts from just transcriptome only to multi-data collections and integration, and that includes epigenetics, which are collected based on using Taxid and Multium platforms, and also spatial transcriptomic. So we have planned these efforts the whole brain level for the mouse, and in a focused area for the human and the macaque. So now going from adults, we're interested in extend both directions in development trajectory. Both teams are interested in understanding developmental mechanisms for cell type identity. For the mouse, we have pretty focused study on postnatal time points with VSP and thalamus. And we have parallel efforts in macaque and human development. And both teams are interested in study aging and neurodegenerative disease. And so we have our whole brain aging projects that focused on RNA-seq using single cell RNA-seq and Multium platforms. And the human study will be focused on Alzheimer's and Parkinson's disease. So next, Amanda will tell you about Multium development, and Kelly, I will talk about aging mouse study, and Rebecca on human disease studies.
Good morning. My name is Amanda Mitchell. I'm a scientist too in mouse genomics, and I will talk about Rajan's nuclei and our multimodal single cell methodologies. So we want to be able to build accurate mouse taxonomies, and most of the data that Chujin discussed was created using cells that are freshly dissected from mouse brains. However, as we move into the future, we'll obtain mouse brain tissue from multiple sources, and we want to be able to utilize frozen tissue. To do so, we need to be able to isolate nuclei. And since mouse nuclei are fragile, many of our current protocols are not able to obtain high quality data. So we've optimized a method called Rajan's nuclei, or ribosome and intact single nuclei, which retains the ER around the exterior of the nucleus. This enables us to retain high gene counts for all of our data. The bottom here, I'm showing you staining with DAPI for the nucleus, S6 for ribosome, and then showing you that we are able to retain our ER around our nuclei. Using the Rajan's method, we can get really high gene counts and can define accurate cell types for both gene expression and epigenomic data. Having both of these data modalities will enable us to create really specific and unique genetic tools for the mouse and both human. To do so, we're using the 10x multiome, which can capture both RNA-seq and ATAC-seq data from the same cell or nucleus. We take our raisins nuclei and they are loaded onto 10x multiome chips. We then are able to process down to both RNA-seq and ATAC-seq libraries. Our RNA-seq libraries are composed of poly-A mRNAs. The libraries are sequenced. The sequencing data comes back and we're able to cluster it based on differential gene expression into unique cell types. These cell types can either be defined directly from the multiome data itself or can be mapped back to the whole brain mouse taxonomy. And then we get specific and unique marker genes for each of our cell types. These cell types are then taken and the labels are directly transferred to our ATAC-seq data. Our ATAC-seq data itself is our epigenomic data. It's the assay for transposase and accessible chromatin. It's basically epigenetic chromatin data. The transposases are basically inserted into open regions of chromatin. These regions of chromatin are then sequenced, and then we basically have open accessible regions of chromatin that are clustered into unique clusters. The labels from RNA-seq data are transferred to our ATAC-seq data, and we can then use this data to obtain unique marker peaks for each of the individual cell types. And these marker peaks can then be utilized downstream to make genetic tools. So the data I'm showing you here all comes from the mouse primary motor area, which we have characterized extensively. But for upcoming projects and grants, we are moving to other regions of the brain. This is showing you striatum data where we are able to also get really high medium gene counts. This orange line is a cutoff of, not a cutoff, but it's basically where 5,000 medium gene counts are. This data, we can then cluster it, we can map it back to the whole brain mouse taxonomy. We've done this for the striatum. We've also done it for other regions, the pallidum, the raphe nuclei, the BTA, and we intend to process this for the entire P56 mouse brain and also use the multiome for other projects that we're currently working on, including the aging mouse brain.
Hi, everyone. My name is Kelly, and I'm a scientist on the mouse genetics team, and I will be talking about cell types in the aging mouse brain. So as Rajan mentioned, we're expanding our efforts from just looking at adult mouse brain into looking at different ages as well. So today I'm going to introduce you to our efforts to profile the aged mouse brain. So as Amanda introduced, we're hoping, or we're in the process of using multiome to profile the entire mouse brain, aged mouse brain. And here I'm showing you a plot looking at gene counts, which is a metric that we commonly use to evaluate quality of our different sequencing libraries across our traditional single cell RNA sequencing modality versus our single nucleus RNA from multiome across the different cell classes, as well as across adult and aged tissue. And you can see that gene counts are comparable between these two modalities, as well as between the two ages, suggesting that we are getting high quality data from both our aged tissue as well as our relatively newer multiome platform. So the aged project is a really big project, and we're collecting data for it in multiple parts. So the first part, which is already complete, we've looked at single cell RNA sequencing profiles across 16 different selected regions across the aged mouse brain. Currently, we are collecting multiome, which is RNA and ATAC-seq profiles across the entire aged whole mouse brain. And eventually we'll also look at select regions using different spatial transcriptome technologies. So for the rest of my portion of this talk, I'm going to focus on the data that we already have, which again is single cell RNA-seq for a select handful of regions. And here are the regions that I was talking about. They fall across a broad range of regions across the brain, including the isocortex, the hippocampus, the hypothalamus, cerebral nuclei, hindbrain, and midbrain. We've profiled both tissues from female and male brains for a total of over 1.2 million age cells so far. So this is a massive data set. Today, I'm only going to focus on a small portion of that data set, specifically the non-neuronal cells, particularly because non-neuronal cells are getting a lot of attention right now in the field of aging research. So to quickly summarize our approach in looking at these cells, what we've done so far is mapped our entire data set to existing internal taxonomies and extracted only the non-neuronal cells. We then performed de novo clustering on both aged and adult cells together to allow them to fall into the same clusters if they look similar transcriptionally. And then with these cluster labels, we can begin to look for age and region-specific differences. So after filtering, this left us with over 650,000 total non-neuronal cells across both adult and age samples. Clustering suggests that there's 152 unique cell type clusters in these non-neuronal cell types alone, which are represented here in this dendrogram. Each leaf node represents a single cluster, and here they're colored by column in terms of which subclass they fall under. We can also color them based on how they break down in terms of the broad brain region that they come from, as well as how they break down in terms of age. If we look at how these cells look in UMAP space, we can see that they group together very nicely by subclass, as is seen here. If we color the same UMAP by what part of the brain they come from, we can see a very striking effect of a regional effect in the astrocyte population here. And finally, if we color by age, at least at this very high level, we can see a very striking effect or separation of age versus adult cells in the oligodendrocyte population. And to focus even more on this specific, on even a more specific group of cells, here we're looking at only oligodendrocytes represented by the 32 different clusters that were found across them. Here they are in their own UMAP space, colored by cluster on the left or age on the right. Similarly to what we saw under the dendrogram, we can also break down each cluster by the proportion of age cells and the proportion in terms of region. Here I've ordered them based on the proportion of age versus adult cells. And using this, there's many different strategies we can take to look for genes associated with age. For example, we can look at clusters that are made up of mostly adult cells versus clusters that are made up of mostly age cells, and look for differential genes between these kinds of clusters. We can also look for clusters that are more balanced between the two different ages and look for genes within these clusters by how they separate by age. And using these kinds of strategies, we can identify many more differentially expressed genes at this very high resolution level compared to if we were to look at the overall subclass. So this has been a very broad overview of one of the many different mouse projects going on. And next, to talk about some of the human projects going on, we have Rebecca Hodge.
Hi, everyone. I'm Rebecca Hodge. I'm an assistant investigator in the Human Genomics program here at the Institute, and I'm going to be telling you today about some of our work in using the techniques that have been described in the previous talks and applying those to understanding human disease. So as you've heard, single-cell genomics techniques are a really powerful framework for understanding and characterizing cellular diversity in the brain. And indeed, we've applied a number of these techniques to human brain, exactly as it has been done in mouse brain. And we've been able to really describe in-depth, high-resolution cell type taxonomies for human cortex, for example. Shown here is a taxonomy of cell types that we described in human middle temporal gyrus that shows that there's roughly the same number of cell types in human cortex, in a single region of human cortex, as there are in mouse. And obviously this is, very useful for understanding all of the different kinds of cells that make up a given brain region. And in addition to that, we can also use this information as a reference for comparison to start to understand disease. So we can use cell type taxonomies to look at identifying cells that might be vulnerable or resistant to disease processes, and looking at gene expression patterns that might be perturbed in association with disease pathology. And so out of that sort of idea of taking these techniques and applying them to disease, the Seattle Alzheimer's Disease Brain Cell Atlas project was born. And this is a large center project that involves a number of different collaborators and colleagues at the University of Washington, Alzheimer's Disease Research Center, the ACT Adult Changes in Thought program, and as well as us here at the Allen Institute running several different projects. And so basically the idea here is that we're going to bring together a number of different types of expertise to really try and understand Alzheimer's disease pathology at a highly granular single cell level. And so I'm going to focus in today on telling you a little bit about our work in this project 2, which is focusing on single cell genomics assays and applications to Alzheimer's disease. So the current goal of the project is focusing on understanding how Alzheimer's disease progresses in the brain. And to get at this, we're looking at doing a population study. And so here we're looking at donors that have low Alzheimer's pathology, all the way up to ranging in severity to high Alzheimer's pathology. And looking across this sort of spectrum of disease, we can start to maybe understand which cell types might be lost or resistant to disease, and what molecular pathways might be perturbed along the course of disease progression. And so we're focusing in on 3 regions of brain that are known to be involved in Alzheimer's disease pathology, and some of which are involved quite early in the disease, including, for example, medial entorhinal cortex and middle temporal gyrus, as well as dorsolateral prefrontal cortex, specifically Brodmann area 9. And so over the course of this past year or so that we've been working on this project, we've focused a lot on generating data and developing a donor cohort. And so right now we have 84 brain donors as part of our donor cohort, sourced from both the Adult Changes in Thought and Alzheimer's Disease Research Center projects. This cohort skews a little bit more toward female versus male donors. And we've been really applying a lot of kind of our workhorse techniques that we use across many projects at the Institute, as you've heard, 10x genomics, single nucleus RNA sequencing to all of these donors, focusing first on middle temporal gyrus. So we have completed data generation for all 84 donors for 10,000 nuclei per donor for single nucleus RNA-seq. And we've also been applying some of these other techniques that you've heard about, like 10x multiome and single nucleus ATAC-seq data to a subset of the donors so far. So that we can sort of get an understanding of how does chromatin accessibility, or how does the epigenome change with the course of disease. And so you can see that, as I mentioned, this is designed to be a population study. And so the donors that we've used in this cohort kind of span the range of Alzheimer's pathology from low to high. This is indicated here by BRAC score, which is a measure of neurofibrillary tangles, or looking at just the overall neuropathological change. You can see that there's quite a range of donors here. And so importantly, this tissue is collected using protocols that were optimized for preserving tissue integrity and RNA quality. And that's been really critical, because many studies of human disease tissue have been challenging to perform because of low tissue quality in the past, where RNA quality was not well preserved. And so with this cohort, we can see really well preserved RNA quality across the majority of the donors, and that's shown here, looking at RNA integrity measurements, where you can see that as you advance in pathology, you start to see a little bit of an indication of lower RNA quality, but generally it's very high overall. We can also look at the quality of the tissue in terms of single nucleus RNA sequencing outcomes, shown here, looking at the number of genes detected per cell, which again, you can see there's a slight decrease with pathology. But overall, we see really good quality sequencing from all of these donors so far. One of the things that we want to look at is trying to understand how proportions of cell types change across donors, across the Alzheimer's pathology spectrum. And you can see here that we see some trends or indications of changes in proportions, for example, here of oligodendrocytes across donors, or layer 5 intratelencephalic projecting cells, which seem to be slightly increased with increasing pathology. But it doesn't seem to be so straightforward, where we're seeing sort of overall loss or change of these cell types reflected in the pathology stage just yet. So more to come on that as we get into more data analysis for this project. So as I mentioned, we're also collecting single nucleus attack-seq data. This is again sort of showing what we've done so far is mostly try to establish that we can use these techniques on aged and Alzheimer's disease donor tissues. Ultimately, obviously, we want to understand how does epigenetic state change with Alzheimer's disease progression. So here we're just showing data from 10 donors. These are donors that had varying Alzheimer's pathology. And essentially what you can see is that right now, using iterative clustering, we can see 71 different cell types with single nucleus attack-seq, which is really comparable to what we see with single nucleus RNA-seq, indicating that this is high quality data. We're able to resolve sort of cell type level information with this data. So next steps in the project is that we're going to be engaging in more data analysis. As I said, we've finished sort of a large chunk of data generation to really try and get at some of these questions of how do cell type proportions and gene expression and chromatin accessibility change with Alzheimer's disease pathology. And next, then we're going to go on to looking at additional brain regions to understand how Alzheimer's progresses across different regions of the brain that are involved at different stages of the pathology. And so just to conclude, what we've shown you today is that epigenomic and transcriptomic single cell and nucleus methods are really powerful tools for characterizing cell types across many different species, stages of development, as well as in aging and disease. And going forward, we are engaging in aligned efforts to really try to characterize cell types across mouse, human, and non-human primate that involve many team members across many departments at the Institute. And so just to close out to say thank you to everyone who is involved in these studies, and thank you for your attention.
Great, thank you for that great talk. And we will look at some of these questions. So our first question is, how much difference do you need to measure in RNA and attack-seq data to decide two cells are different cell types?
So maybe I can quickly address that question. So we have a kind of a pipeline for clustering, and the cell types are determined more or less by driven by differential gene expression. So we decided with two types are separated based on the threshold of how many genes are differentially expressed and how strongly they're differentially expressed. Basically, we're looking for binary genes that have more these on and off gene expression patterns. So by default, I think it's around like 8 to 10 genes. Yeah.
Great. Another question also from Ben is, so the 16 regions for the aged mouse brain represent what % of the entire mass of an aged mouse brain?
I don't know what the exact percentage is, but they are a pretty small proportion of the brain. They were chosen because they reflect regions of the brain that seem to be more affected by age in the literature. We are in the process of profiling the rest of the brain, so there's still a lot in terms of the mass, I guess, of stuff we've looked at. We haven't looked at more stuff than we've looked at, so there's still a lot more data coming in.
Great. Yeah, so here's a question. How do you anticipate following specific cell types on their journey from development through aging? Another way of putting this is, will you need separate taxonomies for all stages, or can they be integrated somehow? Which cell types seem like they are changing most during the process?
So I can answer that question. So the plan is actually, I think we are kind of already have a pilot studies looking at this in the VSP and the thalamus. We plan to build a developmental cell type taxonomy that put together all the time points. So the beauty of our data set is that data all collected in a highly controlled environments. So historically, we don't worry for batch effects. So we can actually put all the cells together and perform clustering analysis and build a one giant, huge joint taxonomy that capture all the cell types at all developmental stages. And we will be able to track their development changes across time, because again, we are not really worried about the batch effects and things like that, and historically. So yeah, that is the plan. We'll see how that works. Yeah.
Great. Question, I think for Rebecca, are all of your Alzheimer's donors dead or some of them still living?
No, the donors are all deceased. So they have agreed to donate their brain at the time of death.
Postmortem, yeah. Also, if the proportion of layer 5 IT types are increasing with Alzheimer's progression, and this holds up as significant, would this be a result of loss of another type or changes in gene expression of another type or more closely resemble another type to more closely resemble layer 5 IT?
Yeah, so those are the kinds of questions that we're hoping to answer sort of going forward with more in-depth analysis of the data. I think, it could be that we'll see changes in certain proportions of types that result from loss of other types in a given layer or in the sample as a whole. As part of the project too, one thing I didn't mention is that there's a major effort to do multiplex fluorescent in situ hybridization to go along with to build off of some of the single cell data that we're generating that will specifically look at trying to quantify changes in proportions in tissue and really hone in on that. So that's a big part of the project going forward.
Charles Laird has a question. Could you please expand on the minimum numbers of transcripts per nucleus measurable with these methods?
I mean, I think that depends on the species that you're looking at. So for example, in human tissue, typically with using 10x RNA sequencing, we see on average around 5,000 genes detected per nucleus. Whereas I know, for mouse, it depends on the methods that you use and can be lower. But
yeah, it also depends on the type. So yeah, with cells, I think sometimes for neurons, we get to up to like 6,000 genes detected for neurons for cells protocol, but for nuclei, it can be lower. I think it's usually in the range of like a 4,000, sometimes 3,000, depends on the protocol we're using and the particular experiments, but this is roughly the range, yeah.
Cool. Here's a question from our NGL Mike Lodato. He says, great talk. Since you see that even when brains are collected under ideal conditions, there are differences in RNA quality between normal and Alzheimer brains. This seems like a feature of the disease. Do you have any thoughts as to how you will bioinformatically deal with that, given that QC steps in many pipelines try to remove poor quality signal?
Yeah, it's a good question. So this is also something that we're, actively looking at in terms of, we want to ensure that we're not removing cells that are of interest simply because they appear to be lower quality as a result of disease. So again, as the analysis of this is really in the early phases of being done, and this is something that we're actively working on right now. So
awesome. And I think our last question, also from one of our NGLs from Fenna, also said, great talk. Can you elaborate on the raisins protocol? Is it mainly beneficial for retaining RNA quality and abundance from frozen nuclei, or does it also help preserve chromatin profiles?
I can answer that question. This is Amanda. So it's mostly just beneficial in retaining RNA quality.
Okay. Great. With that, why don't we wrap up? Thanks again for that wonderful talk on all this great work. And we will move to our next NGL talk, our last NGL talk for this year's showcase. So our last talk is from Kanaka Rajan. She's an assistant professor at the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai in New York. Her talk today is titled How Brain Circuits Function in Health and Disease, Understanding Brain-Wide Current Flow. And she's discussing a new computational framework that she's developed for tracing neural paths across multiple brain regions that enables computation of excitatory and inhibitory currents. She discusses the applications of this technique to the neural underpinnings of behavior in both health and disease, such as depression and anxiety-like behaviors. So welcome, Kanaka. Take it away.
Thank you so much for the kind introduction. It's a delight. I have watched the NGL program thanks to a former NGL and my friend and colleague Denise Tsai with a lot of FOMO. So yeah, I'm delighted to be at least somewhat here. I have big shoes to fill with all the talks that preceded, so I'll do my best. So before I begin, I would like to, thank my colleagues and key collaborators on this project, primarily Matt Perrick, who's starting his own lab pretty soon. The data that I'm going to be talking about today that I build my models based off of was generated in Carl Deisroth's lab. And Tyler Benster and Aaron Andelman are the talented folk in that lab who generated these data. I would also like to thank my funding sources for their kindness and their faith in our ideas. So, we're experiencing this revolution in neuroscience, right? So when I was first starting out in neuroscience, we went from recording single neurons to we were starting to record populations of neurons or groups of neurons within a brain area. But, even today we saw so many talks and I'd like to call out, Lucas Pinto's beautiful talk where we're, thanks to explosive neurotechnological advances and some of which, in fact, the Allen Institute is deeply involved in, we're in this era of unprecedented advances, which allow us to monitor activity across multiple brain regions in awake behaving animals. And one of the things that people have done, and so activity, typically could look like spike trains if you're recording from neuropixel probes brain-wide, for example, as you can see from my, rather poorly rendered version of the inverted hockey stick neuropixels probe. And so, high dimensional data like this are very complicated. And since these data involve signals that are multiplexed across multiple spatial and temporal scales, they're very hard to parse and try to make sense of. One of the techniques, or one of the sort of almost now a field that has come out of trying to make sense of data like this involves dimensionality reduction. So many labs look at state space reductions of these types of high dimensional data. So that involves essentially making a new coordinate frame based on a method like principal component analysis or JPCA or Gaussian process factor analysis, and you get a new coordinate system into which this kind of high dimensional population, potentially even brain-wide activity can be projected. And multiple labs, multiple studies based on this type of data analysis have found that signals that are relevant to performance of different tasks or behaviorally relevant signals seem to inhabit smaller dimensions, smaller than the total n dimensions available to the dynamics. The relevant dynamics seem to inhabit a lower dimensional subspace in this full n-dimensional state space. And then this leads us to the idea of neural manifolds, which are a feature of the covariance or the activity between pairs of neurons in this state space. So the neural manifold can be conceptualized by trying to understand the covariance of this high dimensional activity, and therefore the axes that make up this neural manifold. So this is now currently within the grasp of data analysts, within the grasp of theoretical modelers like myself, and experimentalists as well. One of the things that we don't know, however, or this current conceptualization does not allow us to have, is the idea of what are the inputs that are shaping this covariance, or what are the inputs that shape this neural manifold. So there are a few quantities similar to the one question that I just asked before. There are a few quantities that are inaccessible from measurements or even from current methods of looking at these types of high dimensional electrical activity. And those are, what are the inputs to each neuron from within and across different brain areas? So the thesis that I'm about to develop with you is that the neural manifold is shaped by a combination of inputs and outputs, but the inputs that are shaping these manifolds typically are interactions between brain areas. So even V1 is 7 synapses from the sensory periphery. So we have to look at these inputs as, if I'm looking at a single neuron, or if I'm a single neuron sitting inside this area A cartoon, how much of my activity comes from interactions with other area A neurons, and how much of these of my experience is shaped by interactions with area B or area C neurons. This is not a thing that is tractable by current methods as trivially. We also don't have access to the directionality of these interactions or the effects of common inputs. So covariance matrices can be symmetric. And so do we know if area A, B, and C are reciprocally interacting with one another, or are area A and B interacting unidirectionally with area C, or is area C acting as a common input to both area A and B? Those are not things that we can get at as trivially using current methods of dimensionality reduction or visualization. One other thing that current methods are somewhat limited to are in being able to scale to looking at more than pairwise interactions, or to look at interactions between more than 2 brain regions at once. So one approach that my lab employs, and I'm not the only one, there are multiple labs that take this approach, is to build multi-region RNNs or recurrent neural network models. So when you hear me say the word RNN here, it just means recurrent neural network models in which neurons interact with one another bidirectionally, as opposed to this kind of layered architecture that you might have seen much more commonly. And so to get at mechanisms such as the ones that I just listed before, inaccessible from measurements alone, the approach is to build these multi-region RNNs. And what my lab does is to build them constrained directly by neural and behavioral data. So for example, I would build these network models constrained to directly match the time series activity recorded in one of your labs at the Allen Institute. And I look forward to doing something like that in the future. And so enter this picture, right? So when we build these recurrent neural network models, we of course validate them through a variety of validation techniques involving toy models or ground truth-like data sets. We also look at what happens to these types of networks and analysis methods when you look at small nervous systems in which you can sample a huge amount of the brain, like the larval zebrafish system, about which I'm going to talk a little bit more. And of course, as you go from mouse to macaques to humans, as we've seen in these talks, we are already getting the idea that our ability to sample from these brains drops exponentially as you're going from one nervous system to the other. So one of the things that networks like this allow us to do, and I hope to be able to convince you of this, is that if we have qualitatively similar amounts of data from these nervous systems, we might still be able to extract mechanisms from them that you wouldn't be able to get at from pure measurement. And so, it's not a useless endeavor to be able to, look at a brain from which you're only sampling 0.00001% and not be able to build these models. So I'm about to convince you of something like that. And then once we build these models that are constrained by neural data, we analyze them using some of the same methods that experimentalists such as yourselves will be applying to your data sets, but also some new methods that come out from being able to reverse engineer these types of network models. Because we have built them, we can kind of take them apart to see what made them work in the first place. And we can infer mechanisms that are not easy to get at purely experimentally. So you will see this plot one more time, I think, in my talk, the schematic that you see on the left of my slide. And the mice are red here for obvious reasons, right? I'm looking for collaborators that work on high throughput large scale data from the mouse nervous system. And so with my interactions as an NGL with the Allen Institute, I hope to be able to, have this kind of bidirectional interaction where my tools might be helpful to look at mechanisms in your data sets. And I might be able to use your data sets to validate or in fact make predictions based on them. Right. Okay. So how do we do this, right? How do we build these multi-region RNNs? So it's really quite simple. What we do is to take a single module recurrent neural network model, which involves model neuron-like units that are connected all to all with one another through these reciprocal connections that I've labeled recurrent connections here. And we pretend that each of these networks is now a brain area. So to go from single region RNNs to multi-region RNNs, we just wire multiple ones of them together. So now if I pick the inter-area connections or weights to be sparser or slightly different from the within area connections, then very soon you have this multi-region RNN with a specific structure. So what does this buy you, right? This kind of almost extremely oversimplified conceptualization of a brain area or a neural circuit comes with mathematical convenience, right? It's a convenience to be able to conceptualize the connectivity within this, quote unquote, model neural circuit by means of this directed interaction matrix. So if I were literally modeling individual neurons and the synaptic weights between them, then this thing would be just a synaptic connectivity matrix. But I'm keeping things kind of general because we want to be able to allow for these types of networks to match data that is not necessarily cellular resolution, such as calcium imaging data, such as ECOG arrays, LFPs, and so forth. So this is why we came up with the term directed interactions. So all it is a mathematical structure which contains the pairwise interaction strength of all pairs of units in a network like this. So in the columns are the presynaptic or the sources, and the rows are the postsynaptic or the targets. Now, when you go from a single module RNN to a multi-region RNN, this can take a slightly more interesting form. So for those of us in the room who are fans of matrices and their properties, this structure now becomes somewhat of a block diagonal-like matrix, where within the blocks along the principal diagonal, as you can see here, you get connectivity that is within each area, like RNN A, B, and C, which, if you recall from one slide ago, was supposed to represent the directionality and magnitude and type of interaction within RNN A, B, and C, respectively. But if you look at the blocks that are right next to RNN A and RNN B in this matrix, that tells you the connectivity from B to A and C to A. Now, this three-region network really has 9 submatrices that can tell you all of the within area and inter-area interactions. So except for very, very small nervous systems, we don't have the connectome of any of the complete connectome of any animal. But this type of formalism will be able to at least help us make progress in how to make sense of those types of data sets as they become more and more available. That's the first thing we do, right? We take a single module RNN, we wire multiple ones together to make ourselves a multi-region RNN. Now, if you just started this way and if all the weights were picked randomly, then a network like this would kind of reverberate, sort of randomly and chaotically without any structure that you see in biological data. So the second thing we do is to train the activity of each of these model neuron-like units to match neural data directly. So one of the learning rules that I use is a set target-based recursive least squares, for example, where in the beginning, the activity of each unit I, which is given by this red squiggle here, is matched with a target function that could be derived directly from data or literally data. So that's what the blue trace is. And at every time step, there's a linear error between where the neuron's activation is and where it needs to be. And the entire matrix of connectivity. So remember, I'm kind of building this brain in which what I'm saying is at every time step, the entire brain-wide connectivity is changed in an activity-dependent manner. So while this is why we call these training algorithms and not learning mechanisms, because this is wildly unrealistic in terms of a biological plasticity mechanism, but it is at least a trick to be able to get networks into the right state where they will autonomously, after training, produce activity that they're trained to match. And this is quite powerful. So what does this buy you, right? We did 2 things. We took a single module RNN and we wired multiple ones together to make a multi-region RNN. And 2, we took its activity, which was initially kind of random and all over the place, to match data directly. So that means every neuron has a target function, which is actual data. So once you run the training protocol, what do you arrive at? You get 3 things from a network like this. So assume for the purposes of today's talk, I'm building networks that are as large as the experimental data set. But in my lab, we've played with examples where we've taken a small network and embedded it in a much larger heterogeneous milieu. Or we've done the other exercise where we've taken a very large network and only trained a small fraction of it to match data. But here, for the purposes of today, in the interest of clarity, let's assume every neuron has a partner that is providing it with a target function that it is now supposed to match. So this exercise buys you three things, right? One is that after training, this multi-region RNN produces dynamics that are consistent with the dynamics observed experimentally without any supervision anymore. So the training is stopped, but now you have a full dynamical system that produces the dynamics consistent with the experimental data in perpetuity. So that matrix that you saw, and knowing the matrix of connections and the initial state, you should be able to produce this activity forward. So this is giving you something slightly more than just the covariance. So it's giving you the entire stable dynamical system. The other thing you get from a network like this is the inference of consistent brain-wide interactions. So since we built this network and trained it, we're now able to look under the hood of this network and infer these within and between area connectivity from this matrix that I showed you before. Now, these two things should be fairly obvious because these networks are so powerful that they can kind of match any continuous waveform. So the first one is not a huge surprise. The second one is a bit more surprising. It's actually kind of a cool because you're now able to tell me what are the connectivity motifs within region A, B, and C, as well as between B and A and C and A and B and C bidirectionally that allow for these dynamics to be manifested experimentally. So what I am truly excited about today, and I'm about to show you today, is this completely new idea that we thought of, which is the dot product of those first two things gives you currents due to recurrence. So if you look at just this term that I've written down at the bottom, it gives you a quantity that you couldn't get at experimentally. In fact, you can't get number 2 or 3 experimentally. Right? So what you get here is the currents that are driving, let's say, a neuron A from interactions within RNN A, and how much of its interactions are driven by currents from B or C. The only thing that you're able to observe experimentally are the recorded outputs, not separate whether the outputs are coming from within the brain region or they're originating elsewhere. The currents due to recurrence allows you to separate those things. So what we have, in other words, is a multi-region neural network model where RNN A is region A, RNN B is region B, RNN C is region C. And after fitting it to match the entire time series, you get this directed interaction matrix where in the blocks along the diagonal, you get connectivity within A, B, and C. And if you look at the blocks that are away from the principal diagonal, you get the connectivity from B to A and C to A like this. For the purpose of simplicity, let's just look at the experiential state of RNN A. I'm sorry. So the dot product of these two objects gives you currents due to recurrent interactions, which means that just by looking at the small index J, or in other words, if you go back to this matrix, just by averaging over the right columns, you're now able to figure out how much of the activity that you recorded in region A came from interactions from region A and how much of it were driven by indirect interactions from region B or region C. So this is why, since we're now able to decompose this activity by means of this matrix into these component or source currents, we call this current-based decomposition, or current for short. Now, the combination or the linear or the actual sum of those three component currents still gives you the activity you recorded experimentally. So one of the things that I wanted to show you today was what happens if you applied this method on data from a real nervous system. Now, in larval zebrafish are convenience because you could see through the entire organism. So this is where I want to talk about this experiment for a second that was performed by Aaron Andelman, who was at the time a postdoc in Carl Dijsselraad's lab. So what Aaron Andelman did was to head fix larval zebrafish and give them shocks at these intervals that you see in this highly simplified schematic that I've drawn on top. So as soon as the shocks come in, this is an aversive stimulus for the animal. So the animal whips its tail, making a vigorous evasive movement. But the animal is head fixed, and this is an open loop experiment in that its tail movement doesn't influence the shock times anyway. And so eventually, over time, the animal perceives this as persistent, inescapable stress and stops struggling. So the animal undergoes a phase transition in which it becomes passively coping. So the initial state of behavior in which the animal struggles vigorously to evade the stressful phenomenon is called active coping. And after it's given up and becomes sort of this extreme version of hopelessness or learned helplessness, is called passive coping. Now, active and passive coping, so now the labels that you see on the schematic are a tiny bit misleading, because active coping and passive coping are both adaptive. The onset point is different based on based on whether someone has a susceptibility to clinical depression or not. So this is why this kind of work has high translational potential. In fact, a lot of the work on learned helplessness and active coping and passive coping transitions was done in mice. This is sort of the first time this kind of behavior challenge was captured in larval zebrafish, which allows us much more access. So this is the behavior that I just described to you, quantified. So what you see here is the speed as a function of time, and you can see the time scales over which this happens, right? It's in thousands of seconds. And the light pink bar that I hope you can see on your screens is the time or the duration of the stress or the behavioral challenge paradigm. Now in black is control fish and in blue are the shocked fish. So when the pink bar begins, you can already see the blue goes above the black, and this is averaged over 5 different individuals. And you can see that there's this phase where the animal's tail whips are much more vigorous and pronounced relative to control. But over time, as the stress builds up, the animal lapses into passive coping, and now the blue is well below the black, indicating that the animal has lapped into passive coping. Now, while they're doing this kind of behavior monitoring, the Daiseroth lab is also able to image brain-wide the activity of anywhere between 10 to 40,000 neurons during the process of this entire experiment. So different neurons, as you can see, light up at different time points. They express nuclear localized GCAM, so they're fairly certain that the ROI that they see is actually a cell body that they see. So different neurons light up at different time points in this nervous system. So how do you make sense of these data, right? So how do you take this kind of high dimensional, large scale data set, about 40,000 neurons, and this kind of very slow behavioral transition? So remember, we're not talking about spatial complexity in terms of number of neurons, and we're talking about temporal complexity in terms of how time extended this is. So how do you make sense of what types of mechanisms might be involved in mediating the active and passive coping transition in larval zebrafish? And then there's a next level question, is how much of our understanding from this highly sampled nervous system extends to what we know in mice and direct experiments in other nervous systems where similar phenotypes are observed. So what we did, okay, so there's 2 major findings that they didn't need us for. Right? 2 findings kind of popped out of the experiment. One is that while the animal is undergoing this persistent, inescapable stress experience, there's a steady increase in the activity of the habenula. So what you see here are 2 nuclei of the habenula. This is a locus, this is an area that has been traditionally associated with learned helplessness from mammalian studies. So that's where we wanted to look first. In black is control fish again, and in blue is shocked fish. The dashed line indicate the time of the onset of the first shock stimulus. And you can see that there's this gentle ramp in the habenular, the average activation of the habenula. Habenular hyperactivation has been associated with clinical depression from human studies as well. So this was like a super cool validation. Now, concomitant with this steady increase in the habenular activation, we found a suppression of the activity in the raphe. So raphe is a nucleus that is known to be downstream of the habenula. And its activity decreases slowly as a function of time, as you can see in the blue, which is the shocked fish raphe, again, averaged over 5 different individuals relative to the control fish. And these are the 2 main findings from the experiment itself. So one of the questions we wanted to ask was, well, how do you take these 2 findings and tie them with brain-wide activity? And I mean, are these 2 findings sufficient to explain the downstream cessation of tail movements, even when the stress has not ceased? So one of the things that we wanted to look for is there a cortex in the larval zebrafish system that is responsible for shutting down the movement. So, I'm a computational neuroscientist, so our instinct is to clear the deck and build the simplest possible model, right? So the model that we thought, and I can already tell you this model was all kinds of wrong. The model we thought we were going to build involved Habenula saying, okay, bad stuff's happening, projecting to the raphe, which dumps serotonin into the system and says, I hope you feel better. Now the shocks keep coming and the fish is head fixed, so you're not feeling better despite the serotonin dump. So something somewhere has to be able to shut down movement so you don't go into shock. Now in mammalian studies, this is known to be a part of the prefrontal cortex, like the infralimbic cortex in mammals, that is known to be responsible for shutting down the movement. So that's the area that we wanted to look for, to see if there is a nucleus in the larval zebrafish system that corresponded with shutting down the movement. So in order to identify such a cortex, we tried to build this whole brain model and from within it find a cortex. Now, I don't want to show you the whole brain RNN analysis. Just for simplicity's sake, in the remaining time that I have, I want to show you the results of what happens if you took 3 regions in the larval zebrafish system and analyzed it. So here we build a multi-region RNN, and area A, which is Habenula-like in blue, area C, which is Raphael-like, and then the third nucleus we picked was something that was labeled the telencephalon in the data set. So if I was looking for the cortex, where else would I look for besides a nucleus that looked like telencephalon? The other reason is, if you remember a few slides ago, I motivated this talk by saying we want to be able to disentangle covariances that arise from the effect of common inputs driving 2 RNNs versus true correlations in activity that could arise from interactions between brain areas rather than an external input. So we picked a region that had a lot of neurons in it, and area B in yellow indicates a telencephalon. And this is where they all appear in the brain of the larval zebrafish. And so we build this 3-region RNN model. And this exercise works really well. So those of us who build RNN models, and I think Farzaneh does as well, she's part of the Allen Institute, and I'd be delighted to talk with her after, is that these types of networks work. So here's an example of the output of such an RNN. Here's a Habenula-like RNN unit in blue, Raphael-like RNN unit in red, and a telencephalon-like RNN unit, along with the target function that it was meant to match in gray in all these 3 plots. So and you can see that, yes, the network kind of misses these, sort of high dimensional wiggles, but sort of gets the overall population dynamics fine. We didn't sweat these misfits because, we were looking for dynamics that unfolded over like 45 minutes. So we didn't particularly sweat these misses. Now, just to convince you that I didn't handpick these 3 neurons to show you, we also did state space analysis. Sorry, this should appear. There's like a delay in my click-through. Okay, so then we did state space analysis where we took the activity of all 3 regions and projected them into the dominant principal components. And that's what you see here on the plots on the right. In red is the model's outputs, and in gray are the data. And you can see, yes, again, you're missing sort of these high dimensional wiggles, but you're getting the overall flavor of the dynamics in this kind of state space visualization right. And again, variance explained by the number of principal components. If you look at the number of principal components that capture like 90% of the variance, some idea of dimensionality, which I'm pretty sure Saskia can improve on for us in terms of how we define this dimensionality, we can see that, yes, we're getting like a lot of the principal components. Right, so this matches the activity, right? I already told you this was at best a mild-sized whoop, right? Yes, networks like this are very powerful and they can match the activity. But the thing that I want to emphasize today is this idea of being able to decompose recorded activity into these component currents. And so that's what I'm showing you here. So activity recorded in the habenula you saw showed this kind of ramp. Now, if you use the matrix of connectivity that we extract from the RNNs, you can decompose that activity of 2,200 habenula neurons, for example, into the component currents. And that's what you see here in these pseudo color plots. So you see a habenula to habenula current, raphe to habenula current, and a telencephalon to habenula current. The sum of these 3 still give you the recorded outputs, but you get by looking at the matrix of connections from the RNN, you get one within area connectivity, within area current, and two inter-area currents that are driven external to the habenula. Right? So what I'm about to convince you of today is that this way of conceptualizing activity is actually sometimes a good complement to traditional ways of analyzing data. So, now we can do dimensionality reduction in the current space rather than doing it just on the outputs. So all I'm doing here is doing PCA on the habenula to habenula current, raphe to habenula current, and telencephalon to habenula current. And that gives me a new coordinate frame with axes that are spanned now by the currents. Now, if I take the activity recorded in the habenula and project it into this state space, something interesting happens. You get something that you didn't before. So now what I was hoping would happen is that there would be a ramp in the within habenula currents because we saw the ramp in the habenular activity also. But here I'm taking the times of the shocks, and early shocks are in warm colors and late shocks are in cold colors. And you can see that earlier on in the experiment, it's the raphe to habenula current that is mostly engaged. And it's only later on that habenula to habenula and telencephalon to habenula currents even come on. So, one of the things that I've learned in my years as dabbling between the biological and physical sciences is never trust a 3D plot. So, but before I show you a 2D rendering of this, I want to show you that this kind of inter-area, this time scale separation was invisible if you just look at, correlating the output with behavior as it traditionally done, looking at the activity of a single area and sorting it, or averaging it over different animals. You can get this ramp, but not the fact that the early part came from outside of the habenula. Now I can do state space decomposition of the recorded activity in the habenula into the dominant principal components, and that is certainly helpful because you can see a smooth squiggle in state space, but it's much harder to interpret. Here the axes tell you exactly which component current is being active and when. So this is even more pronounced when I now take this and show you as a function of time. So every time the fish wiggles its tail, it breaks a light beam, which gives you a vector of ones and zeros, which we've now convolved with a Gaussian to give your eye something to look at. So that's what the black trace is in the graph that just appeared. Now in red is the raphe to habenula current, in blue is the habenula to habenula current, and in yellow is the telencephalon to habenula current. And this is an RNN currents from an RNN fit to data from a control fish. Now in shocked fish, the times of the shocks are also put on top. And here you see something completely different in that the early part of the shocks or the active coping where the tail whips are higher than when they cease, that is driven mostly by a ramp in the raphe to habenula currents. And it's only after the fish has lapsed into passivity, as you can see from the tail whips going to zero, is that the habenula to habenula and telencephalon to habenula currents come on. Now, this is not even a particularly great shocked fish because it still waggles its tail after it's supposed to have lapsed into passivity. But this effect is maintained even if you average the outputs of different RNNs based on different individual fish. So this, there's the fact that these three currents kind of mirror one another in control fish versus in shocked fish, where the early part is given by this ramp in the raphe to habenula current. And it's only later on that these other two currents even come on, is maintained even when you look at RNNs that start from different initial conditions and are fit to different fish. So there's multiple sources of variability that nonetheless give you this kind of consistency. Now, to really convince you that there's multiple things going on in the system mechanistically, I also want to bring you back to this matrix of connectivity that you have in these three regions. So here I'm only showing you the histogram of the raphe to habenula projections, because I wanted to understand if this ramp in currents was driven by something intrinsic to that particular submatrix or not. So here I've taken a log of the y-axis, and we've also normalized everybody so the histograms start at 1, so your eye has something to look at and it's not confusing, as a function of interaction strengths. Now the black histogram is the baseline period before any shocks, The red is the early part of the experiment, and the blue is the late part of the experiment. And once again, averaged over multiple RNNs based on different fish. And you can see that early part of the experiment, when the raphe to habenula current is ramping, there's no measurable change in the histogram or the connectivity weights going from the raphe to the habenula. However, it's only later on, almost after passivity, that the raphe to habenula's connections become stronger. So there's multiple different things happening here. There may be slow changes that are driven by changes in brain-wide connectivity, and that you can see by looking at rotations in this kind of manifold that is not the output manifold recorded experimentally, but this kind of current decomposition that you wouldn't have had access to if you didn't build the RNN in the first place. So what have I told you today, right? I've told you two things. One is that habenular interactions do strengthen with persistent inescapable adversity. And we did write that in the other paper. And feedback interactions from the raphe to habenula strengthen, as you saw in the blue histogram. Now today, after we have looked at this kind of current-based decomposition, we can see that there's multiple different things happening in that there's differential roles of the raphe and telencephalon projections into the habenula. Some of these are driven by fast changes to the manifold of the currents, and some are driven by slower, potentially or possibly structural changes. And so we're still trying to understand if there's more than, if there are just these two mechanisms at play, or are there something intermediate between these two mechanisms. But it's this type of approach that I want to be able to apply to the mouse nervous system and disentangle the source or disentangle from multiplexed high dimensional data, whether activity within a brain area is resident within the area or how much of it is inherited from interactions with other brain areas. So with that, I would like to thank you all for your attention and this kind invitation. And I would like to take these discussions forward. Thank you.
Great. Thank you so much for that great talk. We are running a little short on time, but let's just have a couple questions before we move on. And then you can jump into the Q&A section and maybe follow up on a few more. Excuse me. One question is, what predictive power have you demonstrated from this multi-region RNN? For instance, could you train the multi-region RNN on some data and then provide novel stimulation to record new data and see how well it predicts that new data? To what extent have you been able to do that so far?
So in the mouse, not a whole lot, although we've applied this in the methods paper and there's a mouse data set and like that. But let's say in the larval zebrafish system, right? If I make a prediction that there's a raphe to habenular interaction, there isn't one in real life, right? The raphe doesn't project back into the habenula directly. But keep in mind, we then can't take our model super seriously and say, am I going to look for an anatomical monosynaptic projection? Probably not. We're looking for some kind of indirect interaction. So the prediction here would be, if I prevented plasticity in that submatrix or projections going into the habenula, I should eliminate different phases of this behavior. And that is something that, optogenetic investigation in the Dizeroth lab is actually currently testing. So that's one example of this. Of this type of highly sampled nervous system and a highly sampled RNN based on a highly sampled nervous system.
Great. There's a few more questions in the chat box that if you have a chance to go in and answer, that would be great. But I do want to keep us moving so that we don't get too far behind our schedule. So thank you again for that great talk. All right, so for our last talk of today, it's my pleasure to introduce Alan Jones, the president and CEO of the Allen Institute, who's going to be giving a bit of a retrospective on the Showcase and NGL programs. I imagine there's others here who, like me, were around when Alan first announced these two programs, like I think about 8 years ago. And it's really interesting to kind of pause and consider how these programs have developed and grown, and the roles that they play in our institute kind of annual cycle and kind of helping us kind of think about what we're doing and who we are, as well as particularly for the NGL program, its role kind of in the broader scientific community in neuroscience. And so I'm looking forward to hear Alan's thoughts on this as well. So join me in welcoming Alan Jones. Thanks.
All right, looks like I'm live. Well, thank you, Saskia, for the introduction, and thank you to the Showcase Organizing Committee for thinking of me and inviting me. So let me just jump right in. If we're going to talk about the history of Showcase, we do need to go back in the Wayback Machine a little bit. And here's just an overview of the Allen Institute timeline. And I want to take you back probably about halfway through our existence, back to around 2011. We launched the first Showcase and launched the Next Gen Leaders program in 2014. But I wanted to set the stage a little bit to the sort of the why and why we were doing this and why at that time, because I think it's kind of important. So the next slide. So let's go back to 2011. And I think this was a time most people would arguably say brain science was having a moment. And I was feeling it as a sort of external representative of the organization. In 2011, I gave my TED talk on the human brain. We organized a workshop with the Gatsby and the Kavli Institutes at Chichley Hall. The picture is shown here. I'm on the far right here with standing next to Amy Bernard from the Allen Institute. But this was the formative meeting in which the germ of the ultimately what was called the Brain Activity Map white paper came out. February of 2012. President Obama mentioned the brain and a big brain project in his State of the Union address. I was part of this global agenda council for the World Economic Forum. So I was going to Davos and these places. I remember giving a talk at Davos with Henry Markram of the big European effort, Tom Insel of the NIH. And so there was a lot of this in the air. Ultimately, the Obama Brain Initiative was launched in 2013. So this was going around sort of external to the Allen Institute, but internally in the Institute, we were also doing a lot and really moving, I think, at light speed in terms of the organizational development growth. So for that first 8 to 10 years of existence, we largely did these atlases. They were highly regarded, but then Paul Allen wanted more. So we got organized, brought Christoph in, developed a 10-year plan, and ultimately launched that in 2012. This was a great moment for the Institute, highlighted there by this picture of Christoph and Paul. They're looking bored, so they're probably staring at my remarks. And then as if that wasn't enough to get all this activity going for the Allen Institute, as well as representing the Allen Institute around the globe, Paul was pushing me to launch efforts in new areas of biological science. And so I was doing planning meetings and other things for the Allen Institute for Cell Science, which, as you'll see in the next slide, let me hold on just a second. Look at the growth, year-end FTEs. We were also growing and adding lots of great science and scientists over this time. So next slide. So in 2014, we ended the year at 250 people. We were growing fast. We launched that Institute of Cell Science at the end of 2014. At that time, we had an annual budget of 50 million, and we had 4 million in external grants. Compare and contrast that to our 2022 budget, which was just approved by our board. That's a little bit north of 200 million, with something on the order of 50 million in grant funding. So we've continued to grow rapidly. At that time, although we were based in Fremont, we were spread across many different buildings, and that made a lot of things challenging. We had a new building in the works. This picture on the right here shows a bottoming out. So in May of 2014 of our brand new building, where we now call home, except during pandemic time, in which only half of us can get to call it home. So next slide. So during this time of rapid growth, there were, and just the things that were in the air, launching new institutes, efforts, these sorts of things, these themes emerged. One was science really needs leaders. And in particular, science needs leaders that, most of us go into science, and this is myself included, because you love science, right? And it's easy to want to put your blinders on, go after your own problem. And so it's often hard to find people who really want to step above and beyond that, sort of take one for the team, maybe even take one for their own personal passions, to step up and to start to lead in different ways. And I saw this in terms of a very long game as we were building our plan for thinking about future Allen Institutes. And it's, you know, when you get to a certain point, you realize it's all about the long game. So we talked about different ways in which we would do this. One of the things in 2014, we started hiring a bunch of great scientists, and you've heard from several of them here. We know that we're developing great developing talent at the Institute, but we also wanted to grow this network of early career leaders outside the Institute that would become part of our larger family, or Ohana. So next slide, next point. Allen Institute is much more than the Brain Atlas. So one of the things that I found as I moved around the world was that people really didn't know about all the great stuff we were doing. There was, I think, a real desire to start to showcase or highlight more of the great work that we were doing to the larger community. We would do this through annual meetings and these sorts of things, but to have it focused just on our efforts, we thought would be a great idea. Now, one of the things that we also wanted to do here, and this was my own experience as a leader of this organization, as we started to grow, certainly over around 150 people, you add layers of management, you have to, but that breaks down these communication nodes in ways that are very, very challenging in terms of organization, keeping alignment, keeping aligned to mission. We added to that the fact that we were in multiple buildings, so we were ending up having this fragmented culture. We're really trying to find ways in which we could continue to connect in different ways, create synergy across all of our science, and just stay more connected in this way. And so usually when we have these ideas, I certainly go around and I talk to people incessantly about what's on my mind. We gather some steam, get some feedback, and then we laid out something early in 2014, which was, I think, shown on the next slide, which was the original charter for Showcase. So in 2014, we launched, and I'm just going to go through, as I go through the bullets here, I'll say what actually held true and what has actually changed a little bit or morphed. I think all for the good, but I'll comment throughout. So it was to be a 2 to 3 day institute-wide showcase of our science, open to the scientific public. We're not quite institute-wide. This has certainly been mostly focused on brain science. Target dates mid-September annually. At some point in the rhythm of our organization, I think people really opted for wanting to go later in the year. I think they liked it as sort of a capstone to the end of the year. And so it moved to this time frame in December. Next. We wanted talks and posters from all PhD level scientific staff, and in parentheses, no senior management. So the fact that I'm talking to you now means that we violated that rule. But the other part of this, the broadening out of not just PhD level, I loved the RA team talk that I heard. So really broadening this out, and it's career development for everybody to get up and give these talks. It's a lot of work. It's nerve-wracking and requires practice, and everybody did a fantastic job. So another key piece of this, and this is from this leadership standpoint, not run or organized by senior management. I personally felt this way. I know the other senior leaders did as well. This was a great opportunity for the people that are really doing all the work of highlighting their own work and the work that's presented in Showcase. So this was a really important, and I think has worked out really, really well. The initial term was Future Leaders Program, and we wanted this Future Leaders Program participation as part of this evaluative process that we do externally. So next slide. So the Future Leaders Program I think is pretty much held true. With the exception of somebody came up with a much better name than Future Leaders Program, obviously one of the Showcase team or early on, and its intent was to align us with the best young talent and vice versa, aligning our great scientists with those that are out there in the broader world, and partnering, collaborating, building those ties, and building our larger family. It provides a really strong branding for the Allen Institute, I think, and I'm hoping that many people feel, certainly that are next gen leaders, that this is a feather in their cap. I personally would be honored if I was in my developing career and picked by other developing career scientists to be part of this team. We convened the Showcase Organizing Committee early in 2014. The original group created all the rules around this, created the idea how they were going to review, how they were going to select. Next bullet. Each year, the organizing committee were to identify 6 future leaders. We had a lot of discussion around what this sort of early career of future leaders meant. For those of you who know me, I don't like the pyramid scheme that is the postdoc in academia, which says that despite the fact that you've got great training as a PhD, you need to be in some endless training loop as a postdoc. For years, I felt that our leaders are growing right there and they need those opportunities. And so as much as we could identify even people who were that early in their career development to be able to be identified, we thought was important, or people who are just starting in the faculty. 3-year terms, they give their first, the talks in their first year of service, and then ultimately we would have these rotating, and so basically these classes of 18, which we're at now in sort of steady state. Of course, this was in the day when people could come visit. They will come one day early and meet with individual scientists, get an overview of the Institute, and then participate in Showcase. Okay. We also had a key piece of this, which we built on a little bit. This is they'll attend Showcase and we want their feedback. And this is a really important part of everything that the Allen Institute does. We have lots of sort of advisory councils, external views to what we do. And it's always been about show your work, put it out there and let people criticize and get their feedback. It's a jury of your peers from the case of the next gen leaders, but they also participate in these larger advisory councils and other things that we have. So it's a great opportunity for mentoring and cross-fertilization from the larger advisory councils that we have. Okay, next slide. So our first showcase, this is a highlight of pictures of it. You can see here, it was in what we call Fremont Studios. It's I think it was actually, it's a movie studio. So I remember we had it in the middle of the day in the summer, September, but it had a very cave-like bar kind of feel to it, nightclub. You know, this front picture here. You can actually see our board member, Michael Stryker, sitting there. It's I like to joke, it's a Texas Hold'em table. And then the poster session, just overall very well received. I think people were really excited about it. I'm gonna pick on Ben Ouellette here. He just rolled over a big anniversary at the Institute. He was one of our very memorable lightning talk givers. And the next slide, I can show you what, I actually put him in twice because I always enjoyed his lightning talks. And that's one thing that we haven't done during the pandemic, but it's always been one of my favorites. And so this was the next year we were actually in our new building and got to kind of kick off our launch of our new building in the new space. It changed the dynamic, was really nice because we now have an auditorium and a space for posters, etc. Next slide. So click. So my favorites. One of the things that I've loved most about Showcase and Next Gen Leaders is that after this initial, the two slides that I showed you about sort of defining the metes and bounds of it, I really had almost no involvement. That people would inform me, and we would have good discussions around these things if they wanted my feedback. But largely, this was done by our developing career leaders. And I love this because it's been so successful. It just shows get out of their way, let them do what they need to do. I've always, and so again, all those teams inventions, the team talks, which I've also really loved because we're all about team science. I think they're integrated very well. I think they're a hallmark and somewhat different than you'd ever find in any other organizations. I love the lightning talks. I said this before. That was always one of my favorites. So please bring it back, team. Getting to visit posters and talking to teams every year. Somewhat overwhelming, I would say at times, because we would often have limited time to visit all the different posters. And I really did want to see what everybody was doing. I was always very excited about that. But it got harder and harder as there were more people. I've also really loved the watching these networks of the next gen leaders grow. So they're now NGL alumni, and I've heard they, on their own, have started convening at annual meetings like Society for Neuroscience and other things. So, and for all of you NGLs that are watching, keep this in mind, that you're part of the Allen Institute family. And that probably leads me to my final, let's see. So one of the things that I've heard repeatedly from our scientists and even our advisory council members is that our next gen leaders provide some of the best advice to our scientists and our teams, in part because they're a lot closer to the doing of the work than some of our more senior advisors. And so they really provide relevant guidance and advice, and we love it, we appreciate it, and please keep doing it. Okay, so as most of you know, I am moving on from my role as president and CEO. So I'm looking forward to attending Showcase in years to come as a fan. Next bullet. I really want to see the program expanded to the full institute. I think that would be great. And finally, I'd love to see, I mean, part of this whole thing is the long game here. I'd love to see a next gen leader be launching and leading a new Allen Institute at some point in the future. So final slide, just click through here. I always say is that there's so many people that have been involved. I actually asked HR how many people since inception have been associated with the Allen Institute. And it's somewhere around 1,300 people, in addition to everybody else that's been a partner advisor, et cetera. So there's so many people to thank. I have to thank my family for putting up with all the travel that I went globally around in some of their most formative and fun years. But really appreciate it, and really appreciate the time to give you the retrospective. And for all of you, I'll see you on down the road.
Thank you, Alan, for those comments. That was really fun to hear. And thanks everyone who has joined us for this final day of the Showcase Symposium. So this actually concludes the public webinar portion of the events for today. You're welcome, if you have any questions for any of the speakers, to visit the Allen Brain Map Community Forum to submit questions for follow-up with the team talks or the next gen leaders. I think someone will put the link in the chat, so you can continue to keep conversations going there. I want to again thank all of our speakers, our next generation leaders, the team talk presenters, and Alan for all the great talks and participation throughout the program. And I also want to give a big thank you to our communications team, specifically Wilhelmina Geese, Christina Jarvis, and Caitlin Casimo, who've done just a phenomenal job making this event happen and having all of the technology work so smoothly. Finally, I want to thank the Allen Institute founder, Paul Allen, for his vision, encouragement, and support, and whose generosity has made this all possible. For those Allen Institute employees and next gen leaders that are participating in the roundtable discussions, they will start momentarily. You'll need to leave this webinar session and join a separate Zoom link that you have in your calendar invites. So we will see people there. And thanks again to everybody for joining us today. Thanks.